mucous membrane, right? Okay, so some of you do remember. So we talked about mucous membrane being part of the GI tract, right? And mucous membrane also you will find it throughout the respiratory tract, urogenital tract. So it's pretty much similar type of structural or structural difference or similarity you will find. So when you look at, okay, we have to finish the immunology in today's lecture. Then also afternoon I supposed to teach antibiotic resistance. So it might happen that I may have to continue discussing immunology until this afternoon. Then antibiotic resistance may be tomorrow. Okay, so there will be a little bit shuffling and rearrangement, but nonetheless we'll finish everything we plan to do it by Friday, right? That's the goal before you take your exam. So few things just to point out the mucous membrane. As I said, we have a pattern recognition receptors, TLR, and these are like a recognition molecules. The most commonly we call it also PAM. Sorry, PAMs, right? Pathogen associated molecular patterns. So mucous membrane should be able to recognize microorganisms, whoever in that vicinity or in that location, and your immune system should be able to communicate through that pattern recognition molecule, right? So I will explain a little bit more as you go along. And then I also mentioned that the antimicrobials, we had quite a discussion there, that Paneth cell is one of those epithelial cells I showed you earlier that secretes a lot of small peptides, bacteriocin-like antimicrobial peptides. Those are not antibiotics but antimicrobial peptides, and it essentially tries to inactivate pathogens or microorganisms in that environment. So that also contributes to the immunity. And of course, this is some of those examples of antimicrobials: lysozyme, cryptidine. This is another one. This is about 35 amino acid long peptide. Then you will also find, I mentioned about defensin. You heard about defensin? Macrophage, neutrophil, they have these antimicrobial peptides, and those are also very effective in controlling pathogens. And then goblet cell, of course, very important, right? Mucous membrane, without mucus, it doesn't work that well. So you need to have a mucus secretion, and we mentioned that goblet cells, they secrete mucus. Okay, and we also started discussing a little bit about the epithelial barrier damage, right? Leaky gut, and I'm going to talk a little bit more about that as we go along. So essentially, you are disrupting the epithelial barrier in the gut, and so we have microorganisms in the gut, right? At large quantities, large numbers, and you don't want them to cross our epithelial barrier to get to the blood circulation. So that would be definitely a negative impact on our health, including joints and the brain. Okay, so the barrier effect can be in the lungs or in the gut, right? And so as the immunogen or microorganism passes through this system, it activates immune system. Definitely you don't want the immune system to be overactive, and consequences of overactivity is that they are going to produce a lot of inflammatory cytokines, and inflammatory cytokines are not desirable. At low level they are desirable, but as the concentration increases, that necessarily has negative impact on health. Okay, mucus, little bit about the mucus. Mucus is a glycoprotein. Mostly it's actually carbohydrate, right? Let me see if I have a structure of it. No, I don't have. I thought I had one. No, I don't have the structure. So mucus is actually large complex. Okay, it's carbohydrate. But then core of the mucus is called protein called mucin. Okay, and with a hydrogen bond, it forms complex structure. So it's a jelly-like structure, right? And so obviously that's been secreted by the goblet cell, and it has a lot of functions. Instead of talking too much about it, because it has carbohydrate, I mentioned that even if you don't have food for a while, you can still survive. And you survival means your microorganism in the gut. So they can feed on the mucus to survive for extended period of time. Okay? But the mucus, because it's a gel-like structure, it holds all those antimicrobial peptides. A lot of different molecules are present in the mucus. So therefore, mucus is extremely important, right, for our health. So healthy gut means we need to have a healthy mucus layer. Okay, and these are some of those descriptions of the mucus. The molecular weights, like I said, in the millions. Okay, and these cells die pretty quickly because of the programmed cell death, and they will renew every 5 to 7 days. And other aspect of the goblet cell, they respond to microorganisms very well, whether they're pathogenic or non-pathogenic or probiotic organisms, they will play accordingly. So some pathogens may increase the mucus secretion, whereas some pathogen may decrease the mucus secretion. Okay, because that would be advantageous for them if they reduce the mucus secretion, so that they can be invasive, they can go through the system, and sometimes they may stimulate more mucus secretion. So therefore, they may have a upper hand in competing with the natural microflora. Okay, so they do a lot of weird stuff. Okay, but nonetheless, all happens. So the mucus, again, if you study a little bit more, there are 18 genes actually control the mucus secretion or mucus synthesis. One of the genes is called MUC2. So a lot of times we look for the MUC2, which is the secreted form of the mucus. Okay, and we can monitor the secretion of the MUC2 when you are studying, say, effect of probiotics or effect of pathogens or effect of toxins. So these are the way we could monitor it. If you look at the mucus, there are two types of mucus. Okay, again, one is called sticky mucus. Sticky means it attaches to the epithelial cell, and the other one is called loosely attached. And so, this is in the chicken actually. That's the advantage. That's the anatomical classification we use. So this is the front part, like esophagus equivalent to humans, and then the antrum, and then this is almost like a stomach, right, in humans. And then you have a duodenum, jejunum, ileum, colon. And what you are looking at, this dark color is actually the sticky mucus, strongly attached. Okay, and it's very difficult to remove that part of the mucus. Whereas the other one, which is called loosely attached, and if you look at the loosely attached, and then the thickness increases as it goes towards the colonic tissue. Okay, any clue why we have more or thicker mucus in the colon? If you look at the depth, so you can clearly see the loosely attached mucus layer is much thicker in the colonic area. Any kind of guess? Say that again. Oh, HCl secretion. HCl secretion you are talking about here, right? But you are saying less in this area. No, colon we don't have HCl secretion. We get mostly in the stomach. He was saying that because you secrete a lot of hydrochloric acid, and in order to protect our epithelial cells, and we need that. One of the things, the microbial community in the gut, it varies from top to bottom, right? So I think I have one slide. That will give you some indication, because in the colon we deal with a lot of toxic byproducts, okay, because the microbial activity, and also we have a large number of microbes in the lower part of the GI tract. Remember, early part of the GI tract or gastrointestinal tract, we digest food, right? A lot of nutrient absorption happens, and as the food migrates, there are metabolic byproducts which are often toxic, right, that come out as a feces. So in order to protect our body from those toxic byproducts, we need to have a good protective system, right? That will be my first instinct. Like if there are a lot of toxic materials being produced in this area, so that needs to be prevented from absorbing by the epithelial cell. So there will be more problem, right? So that's one way to protect our body against the toxic byproducts produced by a lot of those billions of microbes we have in the lower part of the GI tract. That makes sense? That's like a more general instinctive approach, but technical or the scientifically that make quite a good sense. If you look at the human system, this is again because lack of information from humans. So this is a study, I think 2010, they published it. They're saying that if you look at the mucus layer, the thinner layer again, or the sticky layer, strongly attached, and you can see very thin, and then the colon have a large loosely attached mucus. So this part of the mucus, during infection, your body can lose it, right? You can tell me a couple of diseases where you see mucoid diarrhea, mucoid bloody diarrhea, right? Couple of diseases happens. One is Shigella, you are about dysentery, right? And also the amoebic dysentery. So you see bloody mucoid diarrhea, means a lot of mucus being released from that. So essentially this mucus layer get wiped off, okay? But they try to provide protection. It's a strong barrier against infective agents. Okay, so as I said before briefly, the goblet cell function and the mucus secretion affect severely. So if that secretion changes during innate immunity, because that's the one going to provide large amount of protection, right? And so changes in normal microflora. So if your microflora community changes, then the mucus secretion would be also affected. Harmful enteric pathogens, I already mentioned that. Some of them cause mucus to be decreased, or sometimes it increases. Nutritional or nutrient deficiencies. So like I was mentioning before, if you stop eating or starving, and your mucus layer goes down, okay? And the velar structure also shortens, okay? A lot of things happen. So if you do histopathology, sectioning, you'll figure out that our velar structure doesn't work properly under certain diseases, or especially the nutritional deficiency. It's dramatic. So in order to monitor the effect of either probiotic or nutrient or any kind of positive attributes or same time negative attributes, you could measure the length of the villus. Okay, how tall is it? It can shrink depending on the disease condition. Okay, so these are the factors actually play a role. And in terms of the mucus secretion, it's also, as I mentioned, it controlled or regulated by number of factors, whether it's a microbial toxins or your physiological conditioning, a lot of those factors. So here is an example. Lactobacillus may increase the secretion, whereas the Helicobacter pylori down regulates. Helicobacter pylori, because it likes to live in the stomach, it decreases. And so these are some of those factors you can study, and that affect the mucus secretion. And as I said, mucus provides a tremendous barrier protection. Means it is an important player in innate immune system. Okay. All right. So here, for example, I'll show you the MUC2 I mentioned a few minutes ago. In this case, we are looking at certain function, but you can stain them, and these are individual goblet cells. This is the encoded by MUC2 gene. Okay. So if you want to study that if the mucus secretion is affected by, for example, in this case pathogen Listeria monocytogenes, you don't see much goblet cell. Okay, only few. These are like a cross section of a velar structure, like the finger-like structure I told you. And so if you take a cross section, this is what it looks like, round. And so this is mucus secreting goblet cells. But whereas if you have a probiotic, you can see increased number of goblet cells. So that's a good thing, right? We need higher amount of mucus being secreted by the gut, initiated by probiotic organism. Okay, that's a symbol of health, or say I would say gut health. Okay, this is experimentally you can analyze it. These are some of this. This is also number of MUC2 positive cells you can count, and then you can also graphically present the data. And this is what I was saying. This is the velar structure. And remember, I was talking about the crypt, or where the stem cell are originated. So these epithelial cells, as they move, including epithelial or goblet cells, they all originated from the crypt. They are part of the small intestine or large intestine. Okay, and this length from crypt to this structure, if you are under nutrition deficiency, this will be shrunk. This will be shortened significantly. Okay, and also the crypt depth. If you are healthy, your crypt depth would be much deeper. That's what we call it. But in case you are infected or chronic stress or whatever negative condition may be, the crypt depth would be shorter. And again, technically, you can stain them. In this case, if there is a severe infection, you see this intense color, means the crypt is very active. Right? Why crypt has to be very active? So these are epithelial cells in this structure. This blue, this is the nucleus, right? Everybody remember this structure we talked last time, right? And so what is the thing I was pointing out? If the cells die, they will fall off from the tip, right? So if you see one treatment has a active crypt, this is intense. It took intense staining. Okay, try to follow me. Question I'm asking you. Whereas in this case, the crypt actually not taking intense stain. The staining represent the number of crypt cells. Okay, so if you have a high intensity, you have a more active crypt. Okay, low intensity means crypt or the stem cells are not that active. What is the difference mean to you? Why area in the crypt is highly active? It is making more cells. Why it has to make more cells? Why the body needs to make more cells? Because there are a lot of damage happening somewhere, right? Are you seeing this? Try to understand the logic behind it. So if cells die, that needs to be replaced, right? So these are part of innate system. So they are infected by pathogen, right? So now pathogen come in, they kill these cells. What body is going to do? Your response is going to repair. That's its job, right? So that I don't get permanently disabled. The purpose is to replace those dead cells, because those cells are killed by pathogen. Are you following my logic? So that means if the cells die because of infection, as part of innate system, my task would be, I need to protect my body, I need to protect myself. So what body does is that since those cells are dead, I need to replace those cells, right? Because those cells have certain function in the body. They are absorbing nutrients, right? If your epithelial cells are non-functional, you are not going to absorb nutrients. So you are going to show signs of diarrhea, right? That's what happened. So as a part of the innate system, if the cells die on the top, they needs to be replaced. So in this case, actually this is an infected cell by Listeria monocytogenes, is killing a lot of those cells on the top, and so therefore the stem cell try to make more cells. Are you clear? Not your head, if you understand. If you don't, then I'll try to reinforce the idea. It's very simple, right? It's like more soldiers die in the war, you send more people, right? So essentially that's the same phenomenon. And if the cells are not dying in one case, there is a probiotic happen to be there, it's not killing those cells, so therefore the crypt doesn't need to produce more cells. So the lot of diseases, in order to understand the enteric disease, the gut health we keep on saying, if you kill these cells, like norovirus, you heard about norovirus mediated diarrhea? Norovirus, you heard about it? Or norovirus, or called Norwalk virus, is one of those viruses causes vomiting and diarrhea. It doesn't kill people, but you wish you are dead. That's the feeling. It's like if you suffer from any enteric diseases, a lot of times you go to bathroom so frequently, you feel that's miserable, right? Or you are vomiting because continuously replacing or removing all the fluid from your body. And so norovirus is one of the examples that kills off all of these cells on the top. Okay, a lot of viruses or bacterial pathogens, Salmonella, Campylobacter, E. coli, they can wipe out this lot of this epithelial cells. So your crypt cells would be continuously working to replace those dead cells. It's a part of innate system. So I'm trying to make you understand what is the importance of mucus, right? And then how those mucus secreting goblet cells or our epithelial cell barrier, how they keep us healthy, how they keep us protected. And so our system, these stem cells would be overactive if the cells die on the top. It's kind of replacing all the cells. Okay, so that's the easy way to take it. You can count them. And sometimes also some cells undergo death, like a programmed death, programmed cell death, or necrosis. Also you can measure that. And again, these are part of the innate system to provide protection against infectious diseases. Okay, so what else we have in the gut? So I mentioned that epithelial cells or mucus secreting goblet cells, they provide protection, right? And so if you look at in general, these all together they are going to provide protection. And one of the other tissues, easy to remember, called gut associated lymphoid tissue, or called GALT, G-A-L-T. So there is a GALT, there is a salt, there is a malt, mucus associated lymphoid tissue. So whenever you heard this terminology, easy to remember, think about it, very simple terminology. Gut associated lymphoid tissue. Lymphoid tissue, when you say lymphoid tissue, means it involves lymphocytes, okay, which is part of the adaptive immune system I mentioned last time. So they will be here. So this is actually the communication, or excuse me, the collaboration among the immune cells. So throughout our gastrointestinal tract, we have this in places. Okay. So one is, we talked about antigen presentation last time. You heard about that? Antigen presenting cells. So imagine somebody got in here, or let's talk. Say like if you bring some fruits and vegetables, you bought it from a store, right? And some product you cannot eat it raw, right? Okay, so can I have your attention, please? Okay, so I know everybody has a tendency to drift a little bit this way or that way, if something happening in the room. Anyway, okay, so just let me explain you a little bit briefly. Very simple way. You went to a grocery store, right? You bought some meat, vegetables. I should not say not veg stuff. Maybe say vegetarian stuff, right? So bought some leafy green, okay, and you decided to eat, right? So what are you going to do? You're going to cook, right? So you need to cook it, and then say you have a guest in your house, right? And then you're going to offer that to the guest, right? So you cook it, you offer it, right? And then at the end of the day, you are asking your guest to compliment you how well you cooked, right? Everybody clear? This is a very simple analogy, right? So your guest is happy, and he complimented you, telling you that how well it was prepared, right? And so in turn, what do you think? I feel good, right? I feel strong. I feel mentally, physically feeling strong. So if anything happens, I can defend. So you brought raw material, you cooked, right? You gave it to your guest, whoever came in, he complimented you, you felt good, then now you feel energized. Do you? You feel good, right? If your guests come in and tell you, oh, it's an awful preparation, right? I don't like it. Then you will be very disappointed, right? Are you with me so far? Okay, I like to see you nodding your head. Okay, so what happens in the immune system, exactly same thing happens, right? So if you bring raw materials, like if I catch a bacteria, right? I cannot process it, I cannot eat it right away, okay? So the bacteria needs to be cooked or processed, right? And then in order to get a positive response from my immune system, and then immune system needs to recognize that you are a good host, right? And once you offer that to the immune system, and then in turn they will complement you in the form of, say, antibody or cytokine, okay? That in turn you make you feel good. So what happens here is the collaboration. So if bacteria or pathogens come into the system, right, and then macrophage or dendritic cells, they are going to take that in and they literally cook it, break it down into small pieces, and that's what we call antigen presentation. Okay, or the cells which present it, we call antigen presenting cell. Who do they present? Who their guest is? So let's say helper cell, T cell, T lymphocyte. Okay, and then they are going to collaborate with the B cell, for example. So the T cell can produce some cytokines because they are presented antigen, and then they can in turn activate the macrophage. So you're being a host, you feel now energized, right? And then you can go back and cook more, or if there is a pathogen coming, you can kill it better. Okay, and so the purpose of this lymphoid tissue, essentially raw material or pathogens coming in, they present it to the T cell, and then T cell and B cell communicate. The T cell can produce cytokines that can activate macrophage, they can kill themselves, kill the pathogens. Or if it is a cytotoxic T cell, there is another form of T lymphocytes that can also inactivate. And at the same time, if the B cell is activated, depending on what type of product you brought it from the store, and B cell may think that I can do a better job in helping, so they could produce antibody. So this is the lymphoid tissue. You will see that throughout your immune system, whether in the gut, whether in the mucus, whether in the skin, all of those different places, this lymphoid tissue play a role. Everybody clear? The lymphoid tissue means adaptive immune response. So even though I was talking about innate system, I jumped into the adaptive because this is taking place all the time. And you heard about Peyer's patch? Anybody? Some of you had heard that, right? So the Peyer's patch is one of the localized tissues. You'll find it in the small intestine, and not in the large intestine, mostly in the small intestine. And that's where the M cell is located. And that's where the pathogens can, or microbes can enter the body, and then the immune system can play a role in controlling these. Okay, so this is how the lymphoid tissue look like. And then there are a lot more stuff in the mucous membrane. So we're talking about, these are the different components: lactoferrin, lysozyme, lactoperoxidase, a lot of antimicrobial peptides, as you mentioned. And these are all part of the mucous membrane. Okay. Since you already have the slides, you can read it more. One of the things I mentioned that innate system, we cannot really induce it, right? But we do have a recent trend that we can train them. This is called trained immunity. Okay, and so this is kind of fascinating that if a macrophage recognize a pathogen, right, and then maybe a week later or 2 weeks later, that macrophage again saw the pathogen come in, can it do its job as a first responder? Think about it. Usually macrophage or neutrophil, they will come into the site, they will try to engulf it, they try to destroy it. But before, we never thought that same responder, first responder, may not be able to do the job because they could be inactivated by pathogen. But what we learn now is that some of those macrophages, they can be trained. Okay, and that's where the trained immunity comes. So these are, I think it's a small printout, so may not be able. So some of those naive cells, it could be activated, right? It could be primed, means they already seen that. They could be trained, and they could have a tolerance. So this is kind of like a recent years new concept, saying that immune system can be trained in a way they could do a better job. Okay, I used that analogy before. In case there is a fire, the first responder come in, right? So if the first responders fail, then you look for backup, right? And then backup would be like an adaptive immune system come in and helping. But in this case, those first responder, if they are well trained, they can do much important job even next time when they see it. Okay, so that's a small difference, but this concept actually proved to be very important and in communicating with the immune system and try to prevent it. So there is a resting phase and then the second stimulation, and that's when they become more active. Okay, all right. There are a lot more about this trained immunity. I don't want to spend too much time. All the slides are there, and including the reference. If you want to read up that one, this reference, and it will give you, some resources for your knowledge. Okay. All right. Then I talked about the resident microbes. So resident microbes also major part of the innate system, right? Whether the skin microbes I mentioned earlier, also the gut microbes, microbial community, and you have large populations in your gastrointestinal tract. So we have a bacteria, then we have a fungi, then you have archaea, right? All the yeast. So we have all those different organisms, parasites, also part of our microbial community, viruses also. So they are part of the gut community, and all of them has certain function in the body, and they play an important role in part of the innate immune system. So if I break it down again, I don't want to spend a lot of time. We're talking about the firmicutes, proteobacteria, bacteroides, and large number, right? So whenever you heard about proteobacteria, if you have too many of them, which is undesirable. Most of the pathogens belong to proteobacteria, okay, at the phyla level. Whereas the firmicutes and bacteroides generally very good. You want that population to be enriching your microbial community, and this gives you some numbers. So if you start with the upper part of the GI tract to lower part, so stomach or duodenum, stomach has a high population. Even though we have acids, we still have a microbes in the stomach. Then the small intestine, you will find anywhere up to 10 to the power 8 colony forming unit, or number of organisms, culturable organisms or non-culturable. Ileum and colon, anywhere close to 10 to the power 14 microbial numbers. So we are talking about, very large number of the microbes in the colon area. So remember, now you can link that to mucus we talked about. Because of this such a large number of organisms, plus the metabolic byproducts accumulates in the colonic area, and that's why we need more protection. That's why mucus provides tremendous amount of protection. Okay. All right. So role of the microbiota in immune system is enormous. Okay. For example, they will produce gastrointestinal peptides. They also control endocrine cells. Short chain fatty acids, I mentioned that before, part of the metabolic byproduct. They are also important in the gut health. General function, xenobiotic metabolism, some kind of drug or drug molecules, toxic molecules, microorganisms help repair them. Also angiogenesis, new blood vessel synthesis, microbes help them. Help develop immune system. And then together microflora and immune system are very important. Remember the villus length and crypt depth I mentioned a few minutes ago. These are actually controlled or regulated by healthy microbial community. So now you see this how all are linked together. In immune system, gut microflora. I know we are going to have more discussion or more presentation on this, but then you can see this. These are major part of the innate and adaptive immune system. Okay, there are also a lot of other factors. Again, I want to give you the bigger picture. These microbes play other roles. Okay, but the negative effect of the microbes, we understood that if you have inflammatory bowel disease, right, you have a wrong population of microbes, they may have a negative impact on health. Okay, so those are some of those negative effects. All right, so a diverse community in gut early in life promotes a balanced immune response and prevents allergic diseases, obesity, diabetes. I guess you must have heard about the baby born by C-section, cesarean section versus the natural birth. Already most of you know that difference. So saying that natural birth, you get a lot of microorganisms from the mother that help you establish, colonize a healthy population in the gut early in life. The babies born by cesarean section has mostly a lot of different types of pathogens or microorganisms coming from the environment or the people who are handling, right, hospital environment. So there is a obviously huge difference between the babies who grew up with the natural microbes versus the organisms which are not so healthy population. Okay, and therefore that may dictate type of disease you suffer throughout your life. Okay, and that concept needs to be seriously used or practiced, because this is very important. It's like a wake up in the morning, what do you eat? Breakfast, right? So always you think that the breakfast, healthy breakfast, good breakfast can lead you a beautiful day all day, right? You believe that? So you don't want to get out of home or come to class without a good breakfast, right? Healthy breakfast. Think about the life. If you take the life, if you live 90 years and the 90 years equals to your one day, and how do you want to start your day? Think about it. With healthy, hearty breakfast, that's what we call it, right? And then you will see that your day going very well all the way to the evening and night, right? And so life, you want to start that way. And that can dictate a lot of different diseases. If you follow these recent literatures, the healthy, balanced microbial community makes a huge difference in someone's life. And as I said, a lot of diseases are linked to gut, so how your immune system responds, that makes a big difference. People suffer from asthma, right? Obesity, a lot of those different chronic diseases we suffer from. And a lot of this may start with the microbial community we start with. Okay? Keep that in mind. Again, you are not learning just for the sake of learning. See if you can practice it also. Right? That's the advantage of the students thinking, if I can save myself, right? If I can help myself. Okay, so that's my advice. If you look at the immune system in the mouth, throat, and respiratory tract, if I collect them, all of them together, so we talked about what we have in the mouth. Think about it, right? Saliva. We have a lot of antimicrobials, so lactoferrin, immunoglobulin, and then lysozyme, resident microflora, ciliated cells, that's in the respiratory tract, epithelial cells. So epithelial cells again come back to the how long they live, right? 10 to 12 days, don't forget. So that time also if pathogens attach to epithelial cells, some of the cells has a tendency to slough off, right? The bacteria go with that. And so that's also a immune strategy, right? Coughing, you expel stuff, and alveolar macrophage in the respiratory tract that help prevent pathogens, right? So now if you look at the biofilm in the mouth, right? Think about how your immune system going to work. Okay, so this is essentially in the mouth. We start everything in the mouth, and if you look at of course that's doesn't look healthy, and you can see this inner part of it. You can see a thick biofilm, right? What about this? It's a biofilm, right? Because it's stained with blue, like a crystal violet staining. You learned that in that class, right? Crystal violet staining can stain the carbohydrate in the biofilm, and you can clearly see this is not a healthy teeth. So then you look at the lungs, right? And we talked about the cystic fibrosis lung, because Pseudomonas aeruginosa grow in the lungs. So these are some of those biofilms in the mouth and the respiratory tract, right? So those are the problems there. Then how our immune system going to protect us against those microorganisms? We'll talk about that towards this lecture. Then in the stomach, right? It's pretty common now. You can tell me what we have, right? Acid, resident microbes, then enzymes, and also these are the healthy probiotic organisms, right? One of the conditions called achlorhydria, I think I mentioned that a few times ago. So although we know we have a healthy hydrochloric acid secretion, but some patients, they may not secrete hydrochloric acid. Maybe whatever condition may be. If you are staying up all night long because of the final exams, right, your HCl secretion would be diminished significantly. And that makes you vulnerable to other diseases. So that's also one of the terminology you can see, achlorhydria, and that makes you more vulnerable to infection. Okay, so the HCl is important in its system. In the small intestine, by now you should know the bile salts that play an important role. Especially it kills the gram-positive organisms, right? Then mucus, Paneth cell I mentioned few times, gut-associated lymphoid tissue in the small intestine, and nasal flora. So these are part of the immune system protecting you against the invading pathogens. Large intestine, you're talking of the microflora, good microbial community, and also the mucus as well as the short chain fatty acids. They play immunity. So all of those things are in place to provide protection against pathogens or invading agents. So if you look at the biofilm in the gut, again same mechanism I mentioned, right? It's pretty. So we have in the lumen, we are looking at the mucus layer in the epithelial layer, and you can clearly see this the attachment, microcolony formation, then maturation and dispersion, right? And so the natural microbes in the gut, they are forming microbial community in the form of biofilm, right? And these are healthy, right? This is what we expect. If you have a diverse community, they can provide good protection. But it's not always going to be healthy, right? If we started abusing ourselves with the wrong diet, wrong type of food, or different type of lifestyle, and so pathogens can, or some organisms can colonize, form biofilm, and then can cause chronic diseases. We all agree? So if you look at this, this is the microbial community we talked about, right? And if you have this disorders, so you can see this associated gut disorders associated with biofilm. So we are talking about irritable bowel syndrome, inflammatory bowel disease, peptic ulcer, and achlorhydria I mentioned, gastrointestinal cancers, and these are the type of microorganisms play a role. So we are looking at, we have displaced our healthy microbial community with undesirable pathogenic microbes. Now they are forming biofilm, okay? And once they form biofilm, and they are part of this chronic disorder, right? And so that's what you are going to see. The mucus layer disappears. These organisms become more invasive, right? They continue to irritate your immune system. So there is a influx of inflammatory cells. They're producing a lot of cytokines. So then more immune cells are coming into the site. And they are trying to protect against this invading biofilm. And now you made a complicated disease, which is the inflammatory bowel disease or chronic diseases. That also can contribute to the colorectal cancer, GI cancers, and that also become part of the immune disorder. Okay, and so biofilm play an important role causing those type of diseases. Here are some more examples. Again, these are new articles, and you are saying that if you are looking at the biofilm and you can see the redness, right? These are caused by some of those undesirable microbial community forming biofilm, right? And towards the end, of course, we'll talk about how to get rid of them. And I was using that example during colonoscopy. They can physically wash this or treat with certain antimicrobials or disperse the biofilm and then introduce antimicrobials, okay? Or also quorum sensing inhibitor molecules that can also break down. So number of different approaches people are using in the body to break down this undesirable biofilm, and that way you can help improve the health of the gut. Yeah? Okay. And so the biofilm play an important role in these many chronic diseases. So there are more also examples. I just said therapeutic intervention against GI biofilm. So you want to inhibit the adhesion of these biofilm forming cells. You can also remove the EPS or eradication of mature biofilms. These are the different strategies they use to prevent or take care of this bad microbial biofilm in the body. Okay. All right. So these are other one. I think just said that here. You can also take healthy microbial community, right? The fecal transplantation, which is one of the therapy for C. diff or Clostridium difficile infection, and it's already approved, and I think some cases it's been used. Also, you can use healthy probiotics, right? You can use probiotics to replace the bad organism to help restore your microbial community and get rid of this negative effect of the biofilm. Okay. So this is the organ-wise how the innate system help protect you against whether any infectious disease, chronic disease, or chronic disorders. Okay, and so that's always working in place. So now think about how do the immune system recognize these invading pathogens, right? And one of the way we try to recognize is that, how do I recognize you? Face, right? Your face are this is a feature. Everybody has a certain feature, right? And this is by your face, I know this is you, right? And this is how I recognize you. So now immune cells, they are all sitting there, right? Macrophage, neutrophils, or dendritic cells, all of those cells, or epithelial cells. So the question is, how do they know which pathogen, which are non-pathogen in the body? We got a lot of non-pathogenic microbes, right? What is the best way they can recognize it, right? So they will say, well, he has the face, he got a hair, he got an arm and leg, and all kinds of structures, right? So the immune system try to recognize you by this feature on the individual, on the microbes, right? And so that's why initially we started with called toll-like receptor. We have a receptor for recognizing certain molecules on the cell, right? And these are different types of recognition system and then activates. So if you talk about that called toll-like receptor, initially it was discovered in fruit fly, okay? And what they recognize is that the epithelial cells, they recognize some molecules from the pathogens or microorganisms based on some of the key molecules, key features. So for example, flagella, right? So the epithelial cells can recognize flagella produced by any organism using one specific toll-like receptor or molecule, right? So that's specific. So if I say TLR5, toll-like receptor 5, it always recognizes flagella. One thing it cannot differentiate if flagella come from different organisms. They take it as like all flagella. So any organism with the flagella they help them for movement, right? And then TLR5 would recognize it. That's just the recognition part. Then we'll talk about the signaling cascade. So what it does actually, there are 1 through, I think there are 11, 12, NOD1, NOD2. And so what you are looking at, these different types of cells, whether it's a dendritic cell or monocyte or macrophage, a natural killer cell, or epithelial cells, they have these different types of molecules, okay? And it's telling you that if it is a flagella, TLR5 binds to it, right? If it is LPS, lipopolysaccharide, it's by TLR4. And so TLR2 is usually peptidoglycan or lipoteichoic acid. These are the molecules in the cell wall structure I mentioned. And if you look at the other molecules, let me show you the graphics first. So here think about this is one epithelial cell or macrophage. Okay? So it has this receptor molecule sitting on this. And this is like a PAMP molecule, pathogen associated molecular recognition, right? And so if these molecules are sitting, then it can bind to these specific antigens present on the microbe, right? So this is just the initial recognition. Why we need that initial recognition? Because you need to, if you want to get into a fight with somebody to kill it, right? You need to grab hold of that person also. It's not we are shooting with a gun or some other weapons, right? They will talk about that immune system, how they use it. But in this case, a lot of times it's like a physical hand fight, right? Both of you got angry and you want to, grab his hand and then twist his arm and all kinds of nasty stuff, right, to defeat that person. So essentially, these macrophage or dendritic cells or epithelial cells, they're going to grab hold to that structure. Once they bind to it, and next phase would be that it try to say, maybe you want to swallow it, right, or internalize it. So if it is a phagocytic cell, it will grab, take it inside, and then destroy it. That's the way it works. But the initial recognition starts this way. Everybody clear on this? Okay. Did you notice that some of those TLR molecules are inside the cell? So this is nucleus, nuclear membrane, and you can see this TLR3, 7, 8, 9, and also NOD2 and 1. These are all inside. How does it work? How do they recognize the organism? They are coming from outside, right? So when do the cell use these receptors Are you following my logic? Usually if I'm in the intact cell, right, and my receptors are sticking outside, and if pathogen or microbes come in, so I'm going to make an interaction, right? So now I can grab that and internalize, or I destroy it, right? But some receptors are located inside. How the immune cells using those receptors localized inside the cell? Anyone? How and when they use that? Those are for, not all of them, these are, those who are outside, these are called extracellular bacteria or microbes, right? But they need, some microbes like to get in, right? Intracellular pathogen, right? Or once the epithelial cell, the macrophage or dendritic cell, they bring the antigen inside. Now this is another way of processing them. Now these receptors can bind to, if you look at those where they interact with nuclear material, right? The RNA or DNA, they interact with this. So that means the cell, when they get inside, when they are lysed, their nuclear material can bind to this receptor. Everybody clear on this now? You are bringing them inside, and then now you want to react against those organisms inside the cell. And naturally, a lot of intracellular pathogens there, right? Viruses are intracellular, right? They get in. There are intracellular bacteria also, they get inside the cell. And those organisms can be recognized by this TLR. Okay, now this is recognition. What is next? Now you recognize it, right? You take it in, you break it. Again, it's part of your immune system. You are trying to inactivate those organisms, you don't want it. Okay, so one of the steps of this interaction can lead to activation of signaling pathway. Okay, what is the signaling cascade? What we are looking at is that, what am I going to do? If I'm a macrophage, I grabbed on one pathogen, right? And what step come next? And next step is that, is it going to activate, say this is NF-kappa B pathway. NF-kappa B is, you know, the regulatory pathway, and that leads to activation of cytokine or inflammatory cytokine. Everybody clear? So this is the activation, and when you have these inflammatory cytokines produced by the macrophage, remember the kitchen experiment I was explaining to you, you brought food, you cooked, right? Offered to the, so what we are doing also during the time when you're preparing it, these cells can produce this inflammatory cytokine. And the purpose of the cytokine is that it can activate itself, or it can activate the neighboring cells which are nearby. And they, if they get activated, they can all come in, now destroy the pathogen more effectively. Is it clear? So inflammation, you heard about inflammation, right? And most of the time we talk inflammation is really not desirable, right? But immunological sense, inflammation is an important step. So there are degree of inflammation, you can say, right? So if you have a massive amount of cytokine produced, you can have a massive inflammatory response. That's the bad part. You don't want too much immune cells to come in, and they're going to cause more problem, right? But in this case, inflammation at low level means lower amount of cytokines produced, and that can activate immune system. And then you need that immune cells to come in and help remove those invading microorganisms or toxins, whatever causing that inflammation. Everybody clear? So at low level, so NF-kappa B play an important role. This signaling cascade, the nuclear factor get into the RNA, the DNA, it binds to the promoter, and it activates the cytokine synthesis. Cytokines are proteins, right? Whether it's a TNF alpha, IL-1, 6, name any of the cytokines. Got it? But mostly inflammatory cytokines we are talking about. These are two different. One is called inducible, one is constitutive, means always happens. Other one is the inducible. Microbes, depending on the microbes or the agents, it can do this. Remember, whatever immunology system you study, this is central. This is always going to happen. Okay. All right. So I think that's a good place to stop. Any questions on this? I know, I mean, this is supposed to be the last part of the immunology, but As I said, I think afternoon lecture, I will also continue this so that you get a global picture. So the innate immunity, right? We talked about innate system, what is in place, right? How well they can provide protection against inflammation, or excuse me, invading pathogens or microorganisms or toxins, which is undesirable, right? So do you think this is, we're talking about the negative aspect, right? Negative part of it. Do you think any good thing also induce inflammation? Good, say for example, a good microbial community or probiotic. Well, autoimmune disease, I think you're talking about, that's a little bit different. Okay, there are different aspects of it. But in terms of the inflammation, inflammatory conditions, usually activated by undesirable pathogens, or which is negative. Usually, if you say probiotic, usually it has a called anti-inflammatory response. It doesn't promote inflammation, but there would be certain components. If you take, for example, peptidoglycan, I mentioned that, right? Probiotic has that peptidoglycan. They could have also teichoic acids that also can induce or activate. So it can, some situations that can do it, but the degree of inflammation or inflammatory response is much lower than the pathogen. Okay. So is inflammation desirable or undesirable? We just mentioned, right? So desirable at low level. So it's not bad if you have certain level of inflammation, because you need to recruit other immune cells to get activated, right? And they can perform better. But if you have a severe inflammation, that means we are producing massive amount of undesirable immune cytokines, right? And that can cause severe disease, right? Including septic shock. I mentioned that a lot of times, that the severity or fatality happens because our immune system is overactive, right? COVID is one of the examples. Some people die because of the aggressive response by your immune system, producing large quantities of inflammatory cytokines. So there is a degree, there is a requirement. So if you are doing an experiment in the laboratory, so now you have to define actually if certain treatments showing certain level of inflammatory cytokines, is it good or bad? Because you have to measure it in relation to the overall health of that individual or animal. Okay, and that's how you have to interpret the data. Okay, any questions now? You have the microphone? Oh, no. Any questions before you go for break? You should have a lot of questions. You studied the slides, right? Okay, go ahead. Oh.
So let's suppose there is a biofilm specific antigen. So can we generate memory T cells against it?
Biofilm specific antigen and memory response against it?
Yes, sir.
Sure, you can do that.
Sir, if we can do, then why do immunity fail upon the reinfection?
Is there a chance of reinfection if you do that?
So then why do immunity fails when we got reinfected?
Sure, if you have a memory cells and your memory cells can work against that if you have a reinfection by same pathogen or same microorganisms producing those biofilm-like molecules. It can happen, but then again, all depends on the degree of infection, the severity of infection, how well it's going to work. You got to think about the biofilm. We are looking at the community, right? A structural aspect, and there is we'll examine towards the end. There are different layers, right? How immune cells going to reach to each layer, and that's the most limiting factor. If the immune cells can easily reach to the surface layer, for example, right, which is easily accessible. If the cells are inside, or the exopolymeric substance is there inside, even though you have a memory cells developed against that antigen, but immune cells may not be able to reach because of the physical barrier is already created within the structure. That makes sense? So
Thank
it
you,
will
sir.
work from the surface. So that's why
Okay,
it is effective against individual planktonic cells, and but when it comes to biofilm, it's a whole lot of different game. And that's when it will become, somebody said that before, it will become chronic infection, right? It's not acute. So the best way to get rid of it would be, you need to disperse the biofilm, make it individual cells, and then immune system can work very well. Okay, so it's just the biofilm when it is formed and how effective your immune system would be, that's problematic. And that's when it become chronic disease, means a lot of times these biofilms are formed and they are continuously present, and your immune system try to do a lot of stuff, and they wrap it up, they cover it, they form granuloma, they form cyst-like structure, a lot of those things. These are actually containment of the infection. That answer your question?
thank you, sir.
Okay. Any other question? Oh, there's a question. Go ahead. Oh, you have to run other side.
Mm. So please can you re-explain that part where you explained the toll-like receptors, how they differentiate between probiotics, like good bacteria and bad bacteria, pathogenic bacteria?
That's a really excellent question. So what do you know so far, even though peptidoglycan structure, for example, right, and is present in gram-positive organism or gram-positive organism, but there will be certain molecules or structure that may present something different. But I mean, if you study the probiotic literature, sometimes people complain that some probiotics can induce inflammation. Okay, so probably those probiotic organisms are recognized by immune cells through TLR receptor, like whether the peptidoglycan or lipoteichoic acid or wall-teichoic acids, they will be recognized and they are going to activate immune system. So in that case, you will not be able to differentiate it. But the degree of severity or the response may vary from probiotic to pathogens, definitely. The pathogenic response, you got multifaceted. That recognition followed by immune cells, they're going to produce more stronger response. Because remember, pathogens not only have the structure, they got all those other virulence factors. They got a surface adhesion or pathogenic factors, some of them may be producing certain toxins. All of these different molecules, they are produced by the pathogens. So therefore, your immune system response is going to be much more severe against that compared to probiotic. It's only just maybe peptidoglycan or some of those components, but it doesn't have those pathogenic factors. Okay, and therefore your immune system is not going to aggressively respond to that. So this is how immune system play a role in differentiating this, which is a bad guy versus which is a good guy.
Okay, thank you, sir.
Okay. Any other questions? Okay, looks like you are ready for the break. I think you can have coffee or tea, then that will help you wake up. So come back and we will have a guest speaker here. So we'll have the next lecture at 11:30 or 11:30, maybe a little late. Okay, because I took too much time. Oh, we started late. Yeah, because
Okay, so we are ready with the next lecture. So I formally welcome Professor Krishnan from Laboratory of Structural Microbiology from RCB Faridabad to just introduce the audience about Professor Krishnan. He is a structural biologist that who research articles, reviews book chapters and patents in structural biology. His lab aims to generate structural knowledge by studying the key molecules that establish the initial contact between the host and the microbe. And as you all have learned that in the biofilm process, the first step is the addition of that microbe to a particular substrate. So what are the molecules which are helpful in this addition? So his lab is working on that thing. And majorly he's working on bacterial cell surface adhesions with focus on pilots like structures in gram-positive pathogenic and non-pathogenic pathogenic organisms towards the understanding their architecture and assembly, their role in adherence and the biofilm formation, especially in couple of organisms are Legilactobacillus ruminis, Streptococcus sanguinis, Streptococcus aurelis, that is like more of the dental pathogens, and Lactobacillus rhamnosus GG, which is a well-known probiotic culture. Okay, so with these words, I welcome Professor Krishnan, and for him to about the audience, we are having 56 participants from different universities, Bundelkhand University, then Manipal University, Jaipur, then we have students from our university, Shri Vishwakarma University, Faridabad. So we are having the audience of UG, comprising of UG, PG and PhD students. Okay, so thank you. Thank you, Professor Krishnan.
Yella. Hello. Yeah. Hope I am audible. Hello. Hello. So thank you, Dr. Goyal. And first of all, I thank him for inviting me to this GIAN course on microbiofilm. And this is the first time I am visiting here. The campus looks very nice. And it's me, like he said, like I am Venkatesan Krishna from Regional Center for Biotechnology, and it's an institution of national importance, and it's comes under like Department of Biotechnology, and also it's a Category 2 center of UNESCO Center, and we also provide degree programs and issue certificates, and for our own student and also the affiliated centers, and we also in addition to research regular PhD programs. We have integrated PhD program and also PG diploma in industrial biotechnology, and we also provide 6 months training. So you can look at the website if you are interested. You can check and apply. So I'll be talking about the role of villi from bacteria associated with dental biofilm, basically the oral biofilm dental plaque. I think Professor Arun touched many of these things. Briefly, like the plaque and probiotics, peptidoglycan approach, and surface adhesions. So I am going to connect all of these things. So like he said, like there are like some UG students also. I kept this talk like a very basic level and without too much technical detail. So I'll just try to give only the message. But if you have any question, you can interrupt me in between now. Okay. So let me start with very basic information and background. So what is plaque, right? And it's a kind of thin film that coats on our tooth surface within few minutes after we brush the teeth. And it's a very complex biofilm composed of multiple microbes, and I think more than 500 species you can see in it, but predominantly bacteria. And they can cause gingivitis and periodontitis, caries, and several other oral infections. And they can even like significantly contribute. Of course, like it's a major cause of life-threatening conditions like infective endocarditis, if the bacteria from on the plaque can spread into the bloodstream during the surgical procedures, and also the gum disease. So how does it form? Right? So if you look at its development, it actually begins from the salivary pellicule, a thin layer see here, that coats our tooth surface immediately after we brush teeth within few minutes. And it has multiple components. Sound is not. Too close, I think. Hello? Hello? Okay. Hello? Yeah. We brush the teeth. And it has like a multiple components as you see here, like mucin, proline rich protein, amylase, and so on. There is long list. And these components basically act as attachment sites for bacteria. However, for bacteria, it's not like an easy job, so they can just go there and attach. It has to go through multiple challenges, particularly the host immunological and physical clearances, like Professor Arun said. And for example, particularly in oral cavity, the saliva puts major challenging and dynamic environment. So when the bacteria come into the contact, so they get constantly washed out and we are getting swallowed. We swallow them. So the gut take care of it. So how does it, how are them like attached to this salivary pellicles? So certain bacteria evolve and found a way to quickly attach to this pellicles. And what do? So they express hair-like projections called pili or fimbria to quickly grasp this one of the receptors while they pass through. And these are mostly gram-positive bacteria. And these are called primary colonizers. So this belongs to Streptococcus aureus, sanguinis, even Actinomyces, mitis and mutins and Gorandistec. So once they attach and they provide attachment sites for the secondary colonizers or the late colonizer that we say. And these primary colonizers, they co-aggregate and provide attachment site for the secondary colonizers. And this interbacterial interaction or otherwise the co-aggregation is very, very critical for the biofilm growth. And multiple studies have demonstrated this. And this interbacterial co-aggregation is very, very essential. And like I said, these primary colonizers express a pili called Sates mediated pili to establish the contact with the host surface and also to interact with the other bacteria. So that is what I am going to do it. My lab focus on couple of these primary colonizers and how this assembly happens and that interaction happens. Okay. So before that, I will just give some more background. So what are pili, right? I hope you might have heard or studied in the textbooks. So these are like basically hair-like projections, extends from the bacterial surface, and they have been found like in both in gram-positive and gram-negative bacteria. Until now, like multiple types have been identified. For example, chaperone-Usher pili from E. coli, that pioneering work has been done, and it assembles pili from more than 10 genes involved in this assembly, and it's assembled this pili using like chaperone assisted pili by using the mechanism called donor strand exchange and donor strand complementation. And that E. coli pili very well known in particularly urinary tract infection, particularly in US, it's a very, very big problem. And these people have with the urinary tract infection, they consume cranberry juice. So then later on, the researchers have found that there is a component in that cranberry juice that basically avoids the bacterial attachment to the urinary epithelial ladders or bladder cells, what you call it. And that now there are a lot of components called pilicides or in the clinical trials. So like that type 4 pili, and that involves in twitching motility. and similarly conjugation. So DNA is like it takes DNA through this or exchange the genetic materials through this type 4 pili. And similarly curly pili, and recently there is also a type 4 pili has been identified. And similarly the Sates mediated pili is one among them, but this is mostly found in gram-positive bacteria and particularly more common in primary colonizers. And as you see in the schematic diagram, usually seen in multiple in numbers and with a size of like maybe 20 to 100 Armstrong diameter, and the length can go up to even 1 to 3 microns. And if you look at, as you see here, these are made up of building blocks called pillin. So particularly the Sates mediated pili, there are 3 building blocks of different sizes. So based on the size, location and the copy number, people keep calling them in with different names. For example, the medium size pillin you see here, the major pillin. So with these repeated units of these major pillins put together with on the top of each other in the head to tail manner, and this forms the pillar's backbone, and it usually see multiple in number. So the people call them as major pillins or backbone pillin, soft pillin, and so on. And there is another one which is largest in size, usually located at the tip. So people refer to as tip pillin or pill association. This is the one it attached with the host receptor to establish the contact. And there is another also one, the smallest one, that's called basal pillin. And this connects the assembled pili to the bacterial cell wall. And these genes for these building blocks and also an enzyme called SARTase usually found together in the same place in the genome called Pilus locus or Pilus cluster or pathogenic island. So what this SARTase catalyze the Pilus polymerization, put these building blocks one on that close together one after another, and basically tie up with the covalent bond, so that the whole thing you can assume as a single molecule, because each of these are connected with the covalent bond. So this is the difference between the Sates mediated pili and the other pili. So whereas the other pili are the subunits are connected with non-covalent interaction, whereas this one all of them are connected with the covalent bond. It assumes like a single molecule. So this is very flexible, but at the time it's very stable. So there are these pillins are having like a certain conserved features that facilitate basically the Pilus assembly. For example, they all have signal sequence at the N terminal, and also they have the cell wall sorting signal at the C terminal. So it has the conserved motif called sorting motif. And in addition, the backbone pillin, they all have something called pillin motif. So during the Pilus assembly, so the Sates cuts this sorting motif between the 309 and glycine and links this 309 to the lysine in the pillin motif of next subunit. And this is how that assembly happens. I hope here. Yeah. So this sorting signal peptide at the N terminal and sorting region at the C terminal helps the pillin for the transportation across the transmembrane, and after the secretion they retain on the membrane through their C terminal hydrophobic domain. So as you see here, that actual assembly begins with the tip pillin. So see here, like the Sates recognize the sorting motif at the C terminal and cleaves them between the 309 and glycine, and it forms acyl enzyme intermediate, pillin enzyme intermediate. And this intermediate receive a nucleophilic attack from the lysine, that is the pillin motif. The amino group, the excellent amino group of the lysine side chain. So this attacks resolve this intermediate, and amide bond is formed between the 309 and this lysine. So now the tip pillin is connected to the soft pillin through the covalent bond. So this Sates repeats these cycles, this transpeptidase reactions, and put the soft pillin one after another at the base. So the pilus begins to grow. So once it's returned, reached to a certain level, and that basal pillin is incorporated at the base, and then pilus growth got terminated. And interestingly, the basal pillin is the substrate for another class of surface that is called housekeeping surface, whereas this surface that catalyzes the pillar polymerization is called pillar specific surface or C-type surface. So once the control is transferred to the housekeeping surface, as now the nucleophilic attack comes from the peptidoglycan bridge, and they are covalently anchored on the cell wall. So this surface mediated pili basically the covalent filament, and it's also covalently anchored on the cell wall. So it cannot be like removed from the cell surface just by shaking or by using some detergent, like in the case of other pili. So this is very stable and it cannot be removed easily, and it's very stable. Okay, how does it facilitate the attachment? And there are like extensively it has been studied in various pathogens, for example, group A streptococcus, group B streptococcus, and streptococcus pneumonia, and so on. So from that, the model has been proposed few years ago, how the pili can mediate attachment with the host receptor. As you see here, the pili, the free floating bacteria, extends the pili toward the host surface, and here the adhesion at the pillar's tip and the host receptor basically interact and establish the connection. And this initial attachment leads to the intimate attachment, where the bacterium, distance between the bacterium and the host cell gets reduced. And here other ancillary pillins, even non-fimbrial adhesions, can facilitate these interactions. And once it is done, it recruits other bacteria and it forms the microcolony formation. Of course, like the subsequent stages, like inflammation, I think he explained, and invasion and infection happening further steps. So the critical point is the attachment, the pillite mediated attachment is the first step in the colonization, right? And all the subsequent steps in the pathogenesis or even biofilm highly depends on success of this attachment. So in the case of pathogenic bacteria, so a lot of research has been done like in the last 3-4 decades, how to prevent this attachment. So if you interfere with this attachment, so we can prevent the infections. So for that, we have to understand how bacterium assembles the pili and where it interacts and how does it interact, right? That is what our main goal is. And of course, like if you interfere with this attachment, and it can also serve as one of the possible alternative to the antibiotics, right? So use of which very often results in the development of resistance, and it is a major concern nowadays. So whereas in this anti-adhesive strategies, if you prevent the attachment, that's less likely going to develop the resistance, and because these approaches do not kill bacteria directly, right? So but that it needs the structural knowledge how this interaction or addition process happening at the cell surface level, right? So with this goal in mind, so we picked up that couple of primary colonizers to understand how the pillar assembly took place on the bacterial surface and mediate interaction with the host receptor. Okay. So the first one is Streptococcus aureus, the primary colonizers. One of my students, Rajneesh, worked on this project. As you see, showed in the introduction, the pilus gene, the pilus operon in the Streptococcus aureus is called PI2 pilus, and it has almost like 5 genes, like the PIT A is the tip pillin, and the PIT B is the sharp pillin, and the sort is G1 and G2, we have 2 different sortases, and also it has a CYP A in addition to G1. So when we compare to the pathogens, the pili, sortase mediated pili known in the other pathogenic bacteria, this pili has several unique characteristics. For example, they don't have the basal pili, and also instead of multiple in number on the surface, they express single pilus on the surface. And also this sharp pillin doesn't have the pillin motif. And moreover, the sorting motif in these pillins is not like a canonical LPXTG motif, and they have something different, VTP-TG and VPE-TG. And also, in addition to the sortase, there is also one more gene called CPA. So we don't know what is its role, but studies have, in other pathogen, they have showed that it required for the pillars assembly. And they have predicted it could be kind of signal peptidase like a protein. So then she purified this protein, PIT A and PIT B, and we worked towards the structures to understand its function. So for like she purified the PIT B protein and she crystallized, but again I am cutting down the method details for the simplicity. And she solved the structures, and as you see here, this is the structure of PIT B. And it has two immunoglobulin like fold. And I think all of you know, right, the immunoglobulin, right? So it's a Y-shaped structure. And if you look at that individual domain, so in each of them you have 7 beta strands, like the 4 plus 3 beta strands together, right? And this is what we call immunoglobulin like fold. So all this structure has a similar fold, like you have 4 plus 3 beta strand as you see here, like in this in the topology diagram. But this we refer to as CNAB, as it was first identified in collagen binding protein B repeat region in Staphylococcus aureus. But why we call this as CNAB or IgG reverse fold? Because even though it's made up of 4 plus 3 beta strands, like similar to IgG fold, but if you look at its order of strands, it's in the reverse direction. So that's all we call IgG reverse fold or CNAB fold. But you usually will have a disulfide bridge here in the IgG fold for the stabilization. But the gram-positive bacteria have evolved instead of, because they lack the disulfide bond machinery basically. So instead of that, they form something called isopeptide bond. This can happen between the lysine and the asparagine in the hydrophobic core in the presence of catalytic residue, glutamic acid, or sometimes even aspartic acid. So when they comes together, it's spontaneously formed. It doesn't need any enzyme. So basically, this isopeptide bonds provide stability here in the case of this pillin domain. And these two are like very too extended, and you see here the sorting motif is located over here at the C-terminal tail. And when we try to locate the pillin motif lysine, because it doesn't have a pillin motif, and we cannot look at in the sequence and identify where it is. But when we solve the structures, and then we visually analyzed, and we found it there in the middle of the first N-terminal domain. There is a loop that we call omega loop. It projects away from the core of the beta strands here, and the pillin motif lysine is located over here. And in addition to that, and we have also noticed a proline rich region here, and I listed the sequence here. We don't know what is its role, but studies have speculated that it could be involved in the protein-protein interaction. So then when we looked at the related structures, for example, FIM-A, this is from Actinomyces oris, another primary colonizer, whose structures is known. And here also there is kind of proline rich segment present over here, and there is one N-terminal domain that is missing here. And there is also FIM-T, that is short pillin from again Actinomyces oris type 1 pili. And you see here the pillin motif lysine is located at the interface between the N-terminal domain and the middle domain. But when we looked at our structure, it is located here in the middle of the N-terminal domain at the omega lobe here. And when we compare its related structures, it's like very well structurally conserved here, even though the pillin motif is not recognizable from the sequence, but structurally we could clearly identify this is where the linking lysine is located. So when we looked at the molecules in the crystal lattice, because many of you know, like the crystal actually formed, this is basically the molecules come together and arrange in a periodic manner in all 3 dimensions. So when you look at the molecules in the crystal lattice, and this is how we noticed. So the molecules are stacked on the top of each other, like the way you see here. The tail of each molecule is inserted onto the N-terminal domain. This is the C-terminal domain, D2 domain, or this is the N-terminal domain. They put inserted like a stacked like this. As you zoom it, you can clearly see the C-terminal tail, the sorting motif over here. See, So, the sorting motif, this is a T, up to here we have cloned, and you also the lysine that we have predicted here, the linking lysines. Even though the pillin motif is absent, you could clearly see the linking lysine here, and they are very close to each other within the covalent bonding distance, so that it can form covalent bond. Even in the absence of sortase mediated covalent bond, they naturally come together and stack on each other because of their complementary state. So, then based on that, we have explained how the pillar shaft is formed using the repeated units of pill B. So, the next we looked at the pit A, the tip pillin structures. So, here she has like constructed multiple construct, but again for simplicity and the sake of time, I cut down the technical details. But she purified the full length protein with the thioredoxin DAC, and then she removed the thioredoxin DAC, and also she treated with chymotrypsin. There was one flexible domain to remove that and to improve the crystallization and the diffraction quality of the crystals. And what you see here, the crystals of the full length and the chymotrypsin treated one. And eventually we solved the structures and using a heavy atom called terbium, but again using the technique called sulfur anomalous dispersion, and we call SAD techniques. For sake of time, I am skipping those details, but this is what the full structure, crystal structure we have got. As you see here, it's made up of like 5 domains. There are 4 Ig-like domains that forms the stalk, and there is also some domain that we refer to as VWFA domain, von Weyl-Brand factor A, or integrin domain at the tip over here. So, this I already sold like the CNAB domain, and we have one more new domain here that is called CNAA domain, but this domain is very often called IgG-DE variation domain. Though similar to that, like the IgG fold, this also made like 4 plus 3 beta strands, but you see the additional strands that we refer to as DEA in the IgG fold. So, that is called DE variation domain or CNAA domain, because these domains were like first identified in collagen binding surface adhesion, so that we call it CNAA fold. And interestingly, this CNA fold, it's not like a continuous, it's basically formed 2 different structures of strand. Like as you see here, the N terminal is here, and it makes half of the domain and goes and it forms the D2 domain and comes back and form rest of the domain. So, basically the 2 different beta strands are connected by the isobeptide bond, as you see here. And the top domain is inserted in this domain, as you see here, between the 2 strands here, B and C. So, that is why it's called inserted domain or VWFA domain, that probably mediates the attachment with the host receptor. And here also like the D4 and D5 domains also the isobeptide bond you can clearly see here. And you can clearly see there the N-terminal domain, there is splitted into 2, but connected by the isobeptide bond, it forms a single domain here, and this domain, the VWF domain, is inserted between these 2 regions over here. It's a very complex fold, and even when we try to predict in the first version of alpha fold, and alpha fold cannot predict this fold because it's coming from the 2 different segments. But now the recent version could predict, because we have deposited the structure and alpha fold learn how to do this. So, next, because this is very long and flexible molecule, we make sure like this is the right conformation. So, we also did like a small molecule, small angle X-ray scattering called SAX to confirm what is the conformation in solution. As you see here, there is a pair distribution plot. It clearly shows that there is a elongated curve with a radius of curvature 64 Armstrong, and it's clearly confirmed that it has elongated shape and multiple domains. And the Dmax value is 220, and it clearly matches with the crystal structure over here, and we fitted it in the SAX envelope. It confirmed that in solution also it exists in the elongated conformation, and you can see the fitting over here. So, that the actual experimental scattering and the fitting of scattering curve calculated from the crystal structures. It's very well fit with the good chi-square value. And next we try to understand how does it binds to the host receptor. And here, like I said, that VWF domain or the integrin domain or inserted domain, it's all very well studied in eukaryotic protein. And it's basically, it has a central beta sheet and 3 alpha helices on either side. And there is also a metal binding site at the tip, and there are like a few residues that we call Midas motif, aspartic acid, serine, threonine, and aspartic acid. They coordinate with the metal ion, and it switchbacks to the open and closed conformation. The open conformation is having like high affinity for the ligand. As you see here, for example, in the case of integrin, it binds to collagen, extracellular matrix protein. The glutamate from the collagen, you see a star here, so they directly coordinate with the metal ion. Here it's the magnesium, and this serine and the threonine and another serine directly coordinate, and there is also a aspartic acid here. So when it loses or it goes to the low affinity conformation or closed conformation, you see here that ligand is replaced by the water molecule, and this threonine is pushed back out of the metal coordination here, just comes off, and this aspartic acid comes into the metal coordination. So this is how it goes back and forth, the open and closed conformation, high affinity and low affinity conformation, to facilitate the protein-protein interaction and interact with the extracellular matrix. So we looked at like how does it happen in our structures in the case of PTA tipilin. So when you compare this structure with the PTA, you clearly see the core of the VWF domain is almost same, the central beta sheets and the surrounding helices, right? But we have noticed additional 2 arms. You can see it in the cyan color. So we call them as like inserted arms, arm 1 and arm 2, and that is not there in the integrin I domain. So it looks like this is a new one in this VWF of PTA tip domain. So where does it interact? Because there is no literature information available. So we try to compare again, like is there any pathogenic structures we can compare and get an idea where does it interact. So like there are few structures it's already known. Like for example, this is from the probiotic from our own lab, we have solved it. This is the tip proteins called SPASI from Lactobacillus rhamnosus GG. And there is also 2 structures available, RRGA and GBS104. And these are tipilin from Streptococcus pneumonia and group P Streptococcus. And also there are like MFA5 and PILB. These are like from type 4 pili tipilin, and here is the type 5 pili tipilin. And this is from gingivalis, and this is also from Streptococcus sanguinis. So like you see here, like this is made up of 5 domains and 2 arms, and here it's a 5 domains, 1 arm, and there are 4 domains and 2 arms, and these all have 1 domain, 1 domain. This appear like the PTA is hybrid of all of them. So it has 5 domains similar to probiotic, but it has 2 domains like the pathogenic one. So then we try to compare the tip domains and see whether we could figure it out anything for the binding. So as you see here, the overall core fold is very similar, but we don't have any structural similarity in the case of that inserted region. And the structure based sequence alignment also very clearly indicated that, you can see that arm region and the arm 2 region, you don't have any sequence similarity or identity. And these conserved Midas motif residues are shown here, D, SSS and T and another aspartic. And so this highlighted region belongs to arm 1 and arm 2. So this is very poor sequence similarity. So even we took only the inserted arms and searched against the whole PDP databases and also with these known structures and also the sequence database, but you could clearly see that it is not matching. It doesn't have a structural similarity or sequence similarity. So even we were getting confused, like what to do, how do we figure it out. So then when we looked at these sequences manually, we found that this arm 1 partially matching with the galactose binding signature motif, and I underlined over here. So then we thought probably it acts as a lectin and interact with some mucin proteins. So then we thought like maybe we should do click on microarray experiment to check like where does it bind and what kind of sugars it recognize. So then we have performed this experiment with this kit. It has 100 common glycans used regularly in the experiment and also commonly found. With that, when we performed and compared to the control, we found that it has affinity for galactose and fructose. So then it clearly indicated that probably this inserted arm act as a lectin to recognize these sugar meiosis. But it doesn't belong to any of the known lectin so far has been classified, like a C-type lectin, that lectin, this lectin, because it doesn't have any sequence similarity or even structural similarity with them. So then we thought like to do the co-aggregation assay, because in the literature it's very well known that Streptococcus borealis and Actinomyces borealis interact with one another, and like I mentioned in the introduction, this co-aggregation basically seeds the biofilm growth, promotes, and this is very, very critical. If you stop this interbacterial interaction, it's not growing, the biofilm. So then how does these two bacterium interact together? So we wanted to check this. For this, Rajneesh performed the co-aggregation assay. So for this, basically simply we mixed the S. borealis cells and Actinomyces borealis cells on the cover slice, and we looked at under the microscope. As you see here, this is S. borealis alone, and when we you clearly see the co-aggregations here, and when we mix with the trypsin, basically to shave off the surface protein, and it loses its co-aggregation. And similarly, when we use antibody raised against the whole pit A, the tip plane, and also the pit B, and also the only with the tip domain, and you could clearly see the co-aggregation gets reduced against the antibody of pit A and also the antibody of the tip domain VWFF, but not in the case of pit B, the one that makes the shaft. So it clearly indicates that the tipilin mediates the interaction with Actinomyces borealis, and particularly the tip domain play a role in the co-aggregation. And this also, like the results is very similar when we pre-treated the Actinomyces borealis cell with the recombinant protein that we have. For example, here like VWF protein, when we treated with, it gets reduced. Here also like N-terminal region reduced, but not here when we treat with pit B. So again, it's confirming that the tipilin mediates this interbacterial co-aggregation, and the N-terminal region play a vital role in facilitating this interbacterial interactions. Because we have identified that this inserted arm acts as a novel lectin, so then we check the role of galactose and fructose, and as you see here in the concentration dependent manner, so they inhibit the co-aggregation. And we also check the metal ion, because the VWF has the metal ion, right, the magnesium binding site. So when we removed, also it's getting reduced. And when we incubate that these stains separately, they are not self-aggregate. So the interbacterial interaction is very, very critical for the co-aggregation. So this is what we have proved. So these two interact through the pili mediated, through the PTA, but through the tipilin PTA, particularly the tip domain. And the metal ion and the glycans play a vital role. So then, because this is the first time we have observed the pili mediates the interbacterial interaction, so we wanted to first confirm this before telling this. So Rajneesh has done a pull down assay, like from where does it interact on the Actinomyces borealis. So for that, she used the recombinant purified protein, and she immobilized on the resin, basically the affinity resins, and then she passed through the cell wall fraction of Actinomyces borealis, and she captured the bound protein, and she eluted it, and then we analyzed them in the mass spec. So when we repeated 3 times, consistently we got 2 hits. One is the CAF-A and another is FIM-B from Actinomyces borealis. Basically, this is another primary colonizer. And the FIM-B is basically the tipilin of type 2 pili. And sometimes it is also replaced by the CAF-A. This is basically non-fimbrial adhesion, but it hijacks the pili to sit at the top. So these two are coming here. And like I said, the Actinomyces borealis itself, like it forms 2 different type of pili, and you have 2 pilins, tipilin FIM-Q, soft pilin FIM-P, and the SATAs. and similarly in the case of type 2. And the actomyces uses the type 1 pillage to interact with the host receptor, and you use another type 2 pillage to interact with the oral streptococci. So what we have found is this connection. So then she performed the yeast to hybrid system, as you see here, compared to the positive and negative control. So the direct information between the PTA and the CAFA is very clearly confirmed. But when we did this with the C-terminal region of PTA, we don't see the interaction. So it's again clearly confirmed that the interbacterial interaction happens through the villi, and the tip pillin play a critical role. Even in the tip pillin, the tip domain facilitate these interactions. So based on that, we have proposed a model for interbacterial interaction through the villi, and that is how we have proposed. And that interaction between the Actomyces aureus and Streptococcus aureus is very critical, and this happens through the villi. Both of them assemble the villi, and this pilus tip probably interact with one another, and probably that the VWF domain and the inserted arms recognize probably the glycan. But we need to figure it out where and how does it interact with the host receptor. And in summary, this is how we have proposed from this project. The tip pillin made up of 5 domains, and it has an inserted domain at the tip, and it has inserted arms that act as a novel lectin to recognize glycan moieties on the other bacteria or maybe on the host surface. And we also figured out how the shaft is formed and the role of stabilizing isopeptide bonds. And this is the kind of first report we have showed how a primary colonizer assembled the villi, and with a complete architecture and also the villi mediated interaction the first time. So I think now I switch to the second project. And before that, based on that information, we also proposed a model for how this assembly happens. For this, like we know like how the shaft is formed from the crystal lattice, because these PIT-B molecules stack on one another on the head to tail manner. But how does this tip pillin incorporated on the top, right, at the tip? So for this, we have done a molecular docking. So by treating this T2 domain, basically the D5 C-terminal domain of PTA and D1 domain as a receptor. So as you see here, this is the docked conformation. They are very nicely docked to each other in a similar manner how two PIT-B molecule could interact. Almost in the similar manner, same space. See here, the C-terminal tail of PIT-B molecule over here and the C-terminal tail of the tip domain here. And they are within the covalent bonding distance. So based on that, we have proposed that model. So according to that, again, so I am not going through for sake of time how this assembly happens, like I explained in the first slide. So these components are secreted through the general segregation machinery, retained on the membrane. They recognize the sorting motif and make the sortase pillin intermediate, and the lysine, it receives the nucleophilic attack from the lysine side chain, and the tip pillin is incorporated at the top, and pilus begin to grow. And we also figured out how much rotation and translation happen when the PIT-B repeats along the shaft to form spring-like fiber. So what we don't know right now is what is the role of the CPA here, and why this bacterium needs that for the pilus assembly, but not necessary for the other pathogens. And similarly, how the termination happens, because it doesn't have a basal pillin. So these all need to be studied in the future. So now I will move to the second one, the Streptococcus sanguinis. So like the Streptococcus aurelis, the pilus gene or pilus gene cluster, you have 3 genes here, pill A, B, C. These are basically the pilus genes, and also one sortase that catalyzes the pilus polymerization, and the housekeeping sortase, its gene is located outside the pilus operon, somewhere else on the gene, that basically anchor the assembled pili on the cell wall. And Smita worked on this project. She purified the sortase A that catalyzed the pilus polymerization she removed the signal peptide again and the transmembrane region, and she purified it. As you see here in the gel filtration and the STS page, it's very homogeneous and pure. It's suitable for the crystallization. and she also got the crystals and it diffracted to a very high resolution, and again we solved the structures. And again I am cutting down the too much details, I will go to the message only. As you see here, this is a Saté structures made up of 8 anti-parallel beta strands. You see here, it's all labeled here, and the catalytic triad is located over here. Arginine, cysteine, and histidine. Basically, they form the transpeptidase reaction. And that's written here. And very important thing, what uniquely we have observed here is there is something called lit region between the alpha 2 and alpha 3 connection. I shown here in the pink region and also over here in the topology. Basically, when you don't have a substrate, it occupies the active site cleft here. But what we have observed in a open conformation, because it is away from the active site, this is what we have observed. And this usually have the sequence something called lit motif, but we don't have that motif over here. And I will just explain that in the structure based sequence element in the next slide. And catalytic triad is over here. And one more thing, we have observed a zinc binding to a histidine, which is adjacent to the active site residue. I will explain what is its role in 2 slides. So when we compared all the structures, at least the few structures that has like a open, the closed conformation, you can clearly see here. So these are all other structures, and you see this in the cyan color. This is our sanguineous Saté structures. So all other structures are like occupying the active site cleft, and this one is away from the active site cleft. And this we classify it as a open conformation. So how this has happened? Because this lit motif usually have this DPX motif, and what is the role of this DPX motif? I will just explain in the next slide. And whereas this one doesn't have that motif, whether that is the reason, yeah, that yet we have to figure it out. And the lit is present only in the Satés that catalyze the Pillas polymerization, and this lit is absent in the housekeeping Satés that anchored the assembled pillai on the cell wall. So this lit is specific to the Pillas polymerase Satés. So when we did like a structure based sequence alignment, you can clearly see here the lit. So we don't see the lit motif, the DPX motif in the case of Satés, sanguineous Satés. And this is one of the variable region in the C-type Satés or pillar specific Satés, and you can see the zinc binding histidine is within the active site loop here. So next we compared this, why we have the open conformation, why the lit is away from the active site cleft. So for here, as you see here, like I said, the crystal is like a arrangement of molecules periodically arranged, right? They stack with one another to form a crystal. So when we looked at the structures, there is a beta 7 loop, the bases, the same as this, from a symmetromate or adjacent molecule getting inserted into the active site over here, you see here. And there is isolysine from here, like that interacts with the arginine through the hydrogen bond over here, see. So basically this pushed the loop away. So that's what we have observed, the open conformation. And this insertion into the adjacent molecule cyclically makes, and it makes a dimer like this. So each of this beta 7 loop is inserted into the active site in the cyclic manner. And 2 such dimers like make the hexamer, like in opposite direction, like a screw. And when we align this beta 7-8 loop of pillar specific Satés, and you can clearly see some conserved pattern, like here with the conserved P at the like a second position, basically mimicking the sorting motif LPXTG. So whenever there is no substrate available, this acts as a pseudo loop or pseudo substrate to sit in the active site. So next we checked the Saté structure, pillar specific Saté structure from another primary colonizer, Actinomyces boris, was published recently. So when we looked at its structures, this again is a closed conformation, and it has a typical TPX motif. As you see here, the aspartic acid engages the arginine, active site arginine through the hydrogen bond, and whereas here the tryptophan again making the sulfur aromatic interaction. This is how they engage or occupy the active site cleft when there is no isopeptide. So I mean the substrate availability. And when you compare here the sanguineous sort is in open conformation, so the lit is away, and whereas the symmetry that beta 7 loop comes and inserted here, and then there is isoleucine engages in the with the active site arginine. That's the same position. And there is also complex structure available with the housekeeping sortase and this sorting motif. So when we looked at that structure, you see a substrate here, and here that LPXTG motif leucine is interacting with arginine. So when you superimpose all of them, you could clearly see all three residues, this aspartic acid, this isoleucine, and this leucine all together. And it's clearly confirmed that beta 7 loop is act as the pseudo substrate over here to stabilize the open conformation in the sanguineous sortase, and you can also clearly see here the beta 7 loop is pushed away from the active site, and this is where the nucleophile enters, the secondary group. So next we dock the substrate, the LP and TG in this case present pill A, pill B, pill C in the pillin, and you see here docked conformation here, and we have figured out all the conformation how the basically sortase recognize the substrate. There are this is a 2D diagram, so you can clearly understand how it recognizes the substrate, and there are you see the interaction from beta 6-7 loop and beta 4-5 loop and beta 7-8 loop, and I have classified here all the interactions. This is the hydrogen bonding interaction. It has all type of interaction like hydrophobic charge residues and negative charge residues, and also the polar interactions. And we have identified the subsite or the residues that interact with each of these substrate. We clearly from the sisal bond we basically name it as a P1, P2, P3, P4 position in the peptide, and the corresponding recognizing site we call the subsite S1, S2, S3, and we have identified all the interacting residues with the substrate. So basically, housekeeping sortase located outside the pilus operon generally recognize the basal pillin substrate, and the pilus specific sortase recognize the sorting motif from the tip pillin and the stop pillin for the catalyzing the pilus polymerization. But what we have found in this case, sanguineous, all the pillins, pill A, pill B, pill C, all of them have the same sorting motif, LP and TG. If that's the case, then how this sortase distinguish between the basal pillin substrate and the stop pillin or tip pillin substrate. So this is how this substrate specificity comes. That is what we are working on now, right? Yeah. So next we check the role of metal ion. In the case of staph aureus sortase, the housekeeping sortase, there is a calcium binding site in this loop near the active site, and the studies have shown that the calcium binding enhances the activity of sortase. And in the case of actinomyces voris also they have found, but when we compare this housekeeping sortase with our sortase, we don't see the conserved residues that interact with the calcium ion. So probably it's telling that the calcium is not necessary for activity of this pillar specific sortase in sanguineous. But we have also checked the structural homologue of actinomyces voris sortase, C, that catalyzed the polymerization of type 1 pillars. So there also there was a calcium binding site, but they have showed that it's not required for the sortase activity. But we have identified the zinc, like I showed in the first structure, near to the active site in the histidine here. So when we check the sanguineous sortase, we see that this is basically the zinc is bound between the two molecules at the interface here, and this is what the dimer is made in the kind of trimer and the hexameric conformations. So it looks like this zinc ion stabilizing the intermolecular interactions to form the crystals, but whether it is actually required for the activity, that we wanted to check. For this we have done the activity assay. For this we have designed the fluoropeptide, the sorting motif peptide, basically with the dapsyle and the adons. Basically these are quencher and the fluorophore. When the sortase cleaves between the threonine and glycine, they will get apart and you will give a fluorescent signal. So we measure the activity at this wavelength. So as you see here, the wild type sortase gives a full activity, and we also mutated the active site cysteine to alanine, so it lost the activity as expected. And then we check the effect of metal ion, calcium, magnesium, and also the zinc. And as you see here, calcium and magnesium doesn't make any effect on the activity, but when we incubate with zinc, it completely lost its activity. So then, but again, this is all like a recombinant protein in vitro assay, right? So it has a histidine. Sometimes zinc interact with those histidine and interfere with the activity. So this, we want to rule out that. For that, we have removed the histidine tag. And then we repeated this experiment, again lowering the concentration of zinc from 5 molar to even 1 millimolar. And you can clearly see that 100% inhibiting. And even when with the 0.5 millimolar, you will still see at least 50% of activity is gone. So it's clearly so that the zinc is inhibiting the activity. Maybe this line can be also explored for inhibiting the SARTase activity and prevents pillar polymerization. So the role of metal ion, this is all like, as I showed here, it makes the trimer in the crystal structures, and the zinc is identified between the two molecules at the interface. So we thought probably the zinc mediates the intermolecular contact and makes the trimer and prevents the active site access to the substrate. Maybe that is what it's inhibiting the activity. So just to rule out that option, we checked whether it's forming the trimer in the presence of metal ion. So we checked. This is without metal ion. You clearly see it comes in the monomer. Based on the elution volume, we have checked this, and when we include calcium, magnesium, and zinc, and we don't see any differences in the elution volume. So all of them eluted as monomer. So it clearly shows that the zinc, of course, it stabilizes the molecule and improve the crystal packing to improve the diffraction quality, but it doesn't have any role in making the trimer, but it interferes with SARTase activity. So how does zinc binding affect this activity? That we are still exploring it. So we have mutated that histidine that binds to zinc, and we are repeating the experiment now. So this is how from this study, like I already showed how SARTase catalyzed the polymerization by cutting at the sorting motif and then carrying out the transpeptidase action. So then we can inhibit this SARTase using zinc or finding some inhibitor. So when you cut that, it stops the polymerization. So it will not able to reach the receptor. Okay. So this is one of the approach we can use it. For this purpose, because we have observed the open conformation, we carried out structure-based virtual screening to identify any inhibitor molecules. So for here, we use zinc library through the Schrodinger software, and we followed the standard pipeline, high throughput screening, and we identify some of this molecule, and alpha corrosin was one of them. As you see here, it's clearly sitting at the active site. You see that these are the interactions, and it is one of the top hit, and the routine also like one of the inhibitor for housekeeping SARTase. So we compare with that, and it is better than this. So it's clearly telling that alpha corrosin can be used as a starting candidate to improve further to inhibit this activity of SARTase. So like I said, anti-adhesive approach is one of the promising things that we can use to control the biofilm by inhibiting or interfering with attachment. So what are the possible ways we can interfere with attachment, right? Particularly to stop the Pillay mediated interaction. So first, like either we can use the receptor analogs, like where does it interact? If you know that, you can create analog, so it will interfere with the interaction. Or you can make addition analog, right? For example, here we have identified the VAWF domain interacts with the receptor or the carbohydrates. So in that case, like a inserted arms or VWF domain, you can make it to interfere with the attachment here. Or you can use or synthesize some molecule like from the natural plant derived products, or you can make a small molecule from the chemistry, synthetic material. So that can interfere with the interbacterial interaction. So that is what we have showed. The fucose and galactose can interfere with the interbacterial interactions in the case of PTA here. And similarly, you can use antibody because most of them are immunogenic property, can be used as a vaccine candidates. And we have also demonstrated that the antibodies raised against the PTA or VWF domain interfere with the interbacterial interaction in the same way the host receptor attachment you can prevent it. Or you can interfere with the pilus biogenesis to prevent its pilus polymerization. It's like you disarming the bacteria, so it will not able to grasp the host receptor. So in this case, we are inhibiting the SART-HC, so it will not polymerize. So I have showed like there are multiple ways you can interfere with the attachment. I am showing here like 2-3 approaches that came from our studies by studying the pili and pilus assembly from 2 different primary colonizers. One is to interfering with attachment, another basically cutting the interbacterial interactions, another one to interfere with the pilus assembly. So it can be improved or developed, refined in the futures used as a starting candidate. So with that, I will stop here. And there are multiple people who work on this project. What I presented today is done by Rajneesh and Smita, and she's Rajneesh and this Smita. And also like to thank our previous and present director, Dr. Vinay Salanke, Sudhanshu Vradti, and current director, Dr. Sagu, for providing facilities and funds, and also funding support from DBT, RCB, and DST. And we have central instrumentation facility, X-ray facility, and computational facility at RCB. And most of the crystallography data we have collected at ESRF, European Synchrotron facility. And multiple beamline scientists helped us to collecting the data. But again, I cut down like many technical details because he said like there are UG students, keep it very basic and give only the message. But if you are interested, I can show the data statistics if you would like to go through it. So I'll stop here. If you have any question, I can take. Yeah.
Good afternoon, sir.
Yeah.
Sir, my question is how to make crystal structure and which software is used to make this structure?
Which software I used to make? Yeah, there are n number of software you can use it. What I used is something called CCP4MG. So that's a software developed by a group called CCP4, basically. And if you type in the Google, it will come. And you can also use PyMol. Have you heard about that? You can use PyMol. There are n number of softwares. You can use PyMol, and there is also one more called what is that cryo-EM software that we use? Yeah. Yeah, there are multiple softwares you can use it. And you can check in the paper also. We have mentioned what software we have used.
It's a solid structure, and now we are just here describing the features of the structure, not the process. The process is reduced. Yeah. Yes. Yes.
Yeah.
These are actually small.
In fact, he can take even 1 hour training module. So you have to download the PDP file from the PDP data bank, and you can use one of the software. You can open in the software, and then you adjust all the parameters to make the molecule in the way you want and what you want to show from the molecules. Then you can export into the image file.
After this lunch, actually, I did basic simple lecture here based on this biofilm formation. Biofilm, very basic for UG. But if you guys are interested, then I'll just give a glimpse of how I saw it, because it's a very interesting story. Where not just crystallography, I also used another very less common technique. Very few people are expert for that. I myself learned it from a German scientist, which is called small angle X-ray scattering. And right now, nowadays, cryo-EM, you have heard about cryo-EM is going on. But the cryo-EM also gives you a snap. You have to fit the model here. But in the crystallography, the structure is partially baked, you have to manually bake it. I mean, if the structure contains 1000 amino acids, and after solving the electron density map of just suppose 200 amino acids, then 800 amino acids you have to use your brain and you have to bake it. So it's a very tedious work.
Any other question? Any other question?
Good afternoon, sir. Sir, it's a general question from like which software we can use for simulation, like
For
simulation.
simulation.
Yes.
Okay, there are multiple software again. Like the commonly used one is Amber or Chromax.
Like which can be easily accessible from like
Because
this.
most of them are like a free version. You don't have to pay anything. Chromax you can download. Amber there is like a free version, student version also there. Or it's very minimal fee. You can download and install in the computer and run it.
So we can.
But if your molecule is very big and if you want to run for a longer time, then you need very high-end servers like with the GPU cards. Yeah. And of course, Schrodinger also you can use. Schrodinger, Discovery Studio.
It's a German software, I think.
Okay.
Good afternoon, sir. My question is why the only lysine is cleaved, not any other amino acid in the pili structure?
Lysine. No, not lysine, because it cleaves the sorting motif between threonine and the glycine. But you said like why not other residues, right? And this is what the specificity. This enzyme is designed to recognize only that residue. And you heard about trypsin, chymotrypsin, right? Where do they cleave? Right, after arginine, the lysine, like, so that is for like each enzyme is designed for its objective, like it's a substrate. It has their own substrate where they can receive. That's how like that active site pocket is designed. So they can recognize only that residue. Yeah, that's it.
Okay, thank you, sir.
But you can alter this by mutating that recognizing sites. Any other question?
So no more questions? So thank you, Professor Krishna, for this lecture. It was more on the research oriented, and in the next half we will have another general lecture on the bio. Okay, so I now invite Professor Bhunia just to felicitate our guest, Dr. Sani, Dr. Andresh.
matrix, which we can call extracellular polymeric substance, and these kind of bacteria with the help of this EPS matrix can adhere to biotic and abiotic both kind of surfaces. For example, you can see here some illustrations, like the first one is the biofilm inside a pipe section. You can see this whole cross section is covered, and here you can see in a moving water stream. Here you can see that the surface has the biofilm, and this has been scratched here, and you can see it on the tip of this glass. And here you can see the enamel is highly covered with oral pathogens, the biofilm of the oral pathogens. So a very important question: why do bacteria just want to settle down in a biofilm? It's very simple. Like unity has the strength. So a bacteria alone cannot do anything, but a bacteria in a group can do many things. So here you can see that there are I have highlighted few of the points. The first point is the survival in the harsh environment. You know that bacteria are basically on the surfaces, and surfaces, whether biotic or abiotic, are actually many a times mechanically and physiologically both harsh. So the bacteria, when they are in a cluster covered with the EPS matrix, they are actually having some survival benefit. That's why they make this bacterial biofilm. Then another is a nutrient trapping and storage. So sometimes the nutrient molecule can be bigger than the bacterial cell itself. So if the bacteria combine together into a giant structure, then they can trap molecules which are nutrient bigger than their size, and can use it for their nutrition for a longer time period. They can have quorum sensing, because quorum sensing, basically, if I tell in one line, is basically the communication between two bacterial cells or more or among the bacterial cells. So this actually helps them to organize into certain way. And then there are, if the bacteria have this matrix, they have a stable surface attachment. Why this stable surface attachment is required? It's very important, because the bacteria, let us divide bacteria into two simple categories: good bacteria and bad bacteria, or pathogenic or probiotics. So irrespective whether they are pathogenic or probiotic, they can show their virulence or the good effect only when their membrane is bigger, because bigger is the membrane, bigger will be their effects. So that's why stable surface attachment is the crucial point. Again, there is a facilitated gene exchange. You may have heard about the horizontal gene transfer. So bacteria which are not actually pathogenic, sometimes are less pathogenic, can transfer their genes to the nearby bacteria and make them pathogenic. Protection from antibiotics and host immune system, because these are the biggest enemies for the bacterial cells. Either the immune system of the host or the antibiotics that we use to kill them. So what happens, I'll discuss it in upcoming slides, when you will see that how the matrix helps them to combat these enemies in their language. So here is a quick revision of how the bacterial biofilms develop. So when the bacterial cells are single cells, we call them planktonic cells. Planktonic cells, why? Because they are free to move anywhere. What happens when a planktonic, group of some planktonic cells adhere to a surface in a reversible attachment manner? Actually, we can divide this whole into two parts: reversible attachment and irreversible attachment. So what happens? The initial attachment of those planktonic bacteria on the surface is actually reversible. But over the time, they secrete several molecules. Some secretions are there from their cells, and that actually covers them, which you can see here as a yellow covering. That is basically that EPS, the matrix. And the moment this matrix comes into the picture, this attachment becomes irreversible. Now what happens? Bacteria multiply and they grow into clusters. When these clusters grow into bigger colonies, what happens? It's actually simple physics. What happens? When they grow, their mechanical stress cannot, the mechanical forces cannot hold that bigger structure into that, because the attachment at the surface is smaller. So simply what happens, these microcolonies get disturbed or perturbed, and what happens? They break down. And these again become free, and now they go to another location and infect there. This is how this infection increases along with the time. So this is the classical first 5-step model, which was basically described for the first time using structural biology techniques for this Pseudomonas aeruginosa. But the question arises that, okay, bacteria adheres to a surface, but how? Does bacteria have hands, legs, or what? So what happened? The bacteria evolved a special structure over their surface that we can call pili, or sometimes we call them fimbri. Note this fimbri is different from that flagellal structure. So what happens? This most of the time this pili or fimbri are scattered throughout their surface, and this is the organ which actually helps them to adhere. Here I have, for example, I have here shown in the first diagram the this electron micrograph of the pilus. Pilus in a probiotic bacteria, for example, Corynebacterium diphtheriae, and here in the second panel you can see the pilus structure in a probiotic strain Lactobacillus rhamnosus GG, which is called the common probiotic. It's a very So let us go a little deeper. What happens when we observe this pilus closely? We find that this pilus is actually made up of 3 subunits. The first one, which is at the top, is called tip pilin. The bottom one has basal pilin, and there are multiple copies of backbone pilin or shaft pilins. How these numbers are decided, how the length is decided, is the another story. So I'll try to tell you in the second half of my presentation. But here you should notice that the virulence of the pathogenic bacteria or the health benefits of the good bacteria is directly proportional to the formation of the biofilm. Bigger the biofilm, bigger will be their effects. So let us closely see this structure APS. What does it compose of? It is actually called a house of biofilm cells because it is the structure where bacteria reside. And what are the components? It's actually made up of 85% matrix and only 15% are bacterial cells. Out of that 85%, 90% is water. And the remaining portions, here you can see basically there are different types of sugars. 1 or 2%, there are several enzymes that bacteria actually release for their defense and for nutritional uptake. And there are certain structural proteins which actually maintains this colony, and there are certain DNA which is DNA molecules. Here you can see which actually are the part of that horizontal gene transfer mechanism. So what regulates this biofilm development? There is a detailed chemistry behind it, but I'll try to give you the overview. So there is a molecule called cyclic di-GMP. So what happens when the number of cyclic di-GMP increases? You see here, when it increases, then it inhibits the motility and increases the adhesins. So what happens whenever the cyclic di-GMP increases, bacteria leave their planktonic stage and they start attachment to the surface and they grow into biofilm. But when the signal comes to decrease the cyclic di-GMP, so what happens? The reverse effect happens, and now the motility and the virulence is favored, and the bacteria again convert into the planktonic. That's where we say that the microcolonies actually break down and the bacteria goes to infect other places. So I will not go into detail, but a few points I would like to highlight here. So here you can see this is the motile lifestyle when the bacteria are planktonic, and here you can see the biofilm in case of inside this matrix. So what happens? This is the cyclic di-GMP. When it is low, it is favoring this planktonic stage, and when high, this favors this one. So what happens? There are several enzymes. Like here you can see guanylate cyclase enzyme, which actually converts cyclic di-GMP which actually synthesizes the cyclic di-GMP from 2 GTP molecules, and this is how the number increases, and where their number increases, this favors the biofilm formation. But at the same time, when the signal is not in the favor of biofilm formation, then there are two phosphodiesterases. The phosphodiesterase 1 actually linearizes this cyclic GMP and converts it into this 3 to 5 prime phosphodiester, creates a 3 to 5 prime phosphodiester bond, and thus it forms a linear dinucleotide. And this linear dinucleotide actually degrades to GMP, and this is how the GMP's number decreases. So since there are biofilms, so are there some techniques to actually detect them or to see them? So right now, two major techniques that are being used throughout the world, which are the confocal laser scanning microscope and the electron microscope. So here in the first diagram, here you can see that there some portions are green and some portions are in red. So what happened in case of this study, they actually grew the bacteria on a plastic surface for a week, and they then stained it using two dyes. The one dye, which is EPS, is responsible for the green color, and it actually binds to the polysaccharide in the EPS. And the bacteria here are shown in the red color because the propodium iodide actually binds to the bacterial DNA. So when they use the confocal laser scanning microscope, you can clearly see here the biofilm. And another major technique that has been used for the visualization of this biofilm is the scanning electron microscope. Same study, same way, but the bacteria were grown on a surface of plastic. This was actually polyethylene glycol, polyethylene, and they visualized is using this electron microscope. So here you can see this biofilm. Now I'll quickly jump to section number 2, where I'll discuss the pathogenicity and clinical relevance. I'm not going into the mechanical part. I'll just highlight that bacteria cause diseases. So if we go here, so probably all the body parts, whether internal or external, which are exposed to external environment, like our gut, when we eat food, so it is getting exposed to the external environment, and similarly this dermis. So all the body parts which are actually getting exposed directly or indirectly have been found to develop biofilms. And these biofilms, I have just listed few of the organism depending upon the different body organ systems. But the important thing is that these bacteria not just even cause simple infection or some inflammation. Some of them are responsible for causing cancer also. So it's a big threat. So that's what I have highlighted. I'm not going into detail. So just highlighted that they can be actually serious threat to life. And you may have heard that most of the people who go to hospitals, most of the time they encounter these infections, bacterial infections. Why? Because there are certain devices which are used in case of medical treatment. For example, in the orthopedic implants, these syringes and this cannula, or this is called this catheters, urinary catheters, or the lenses even, or the cardiovascular implants, like the different kind of implants are there. So all of them are also exposed to somewhere, or either open surfaces directly, indirectly, or to the blood. And this bacteria which migrates from one place to another after this disruption of that biofilm can actually form a biofilm on these devices also. So these are also cause of increasing the infection in case of not just patient but the health caretaker and also those who visit them. I have just taken two examples to show how they cause the harm. For example, here I have taken a very simple example of dental carie. What happens when the this upper surface of the this teeth is called enamel. So whenever the bacteria, this plaque actually, which is a bacterial biofilm, grows here, what happens is they secrete different types of acids, and they secrete those acids in order to dissolve or digest their food outside their body. But here when they release those acids, what happens, this enamel covering of the teeth actually, which is very hard, but it gets actually corroded over the time, and the dental caries takes, and once the dental caries, if not treated, reaches to the vascular system, then it can migrate to other body parts also. I have not shown here, but in one of the study has been shown that this dental plaque was actually causing heart diseases. This is how you can see, because once they are exposed to bloodstream, they can go to anywhere in the body and they can infect anywhere. So you should have oral hygiene, because it's not just for your oral cavity, it's for your whole body, because it's very difficult to detect the organisms who are actually the component of those particular biofilms. Another very dangerous example I'll give here is about the cystic fibrosis lung. So if you see the normal lungs, what happens if you go to their alveoli? So what happens basically on their inside the alveoli, there are basically a thin layer of the mucus covering the lung epithelium. So that's why the lung epitheliums are not actually disturbed through these. Actually, whenever we inhale or exhale, what happens is there are different body parts which actually try to clean that air. But what happens if you are living in highly polluted area like Delhi? So it's not possible. That's why people living in Delhi or any highly air pollution area actually develop many these respiratory diseases. One of them can be because of bacterial growth also. Here you can see what happens when the bacteria starts growing here. Suppose it went through your nostril, through your respiratory system, and it grows here. What happens? This actually starts disturbing this mucus layer, and it gets exposed to the epithelium of alveoli. And just think, if it is exposed to the epithelium of the alveoli, then obviously the oxygen intake will be oxygen intake inside the bloodstream will go down, and this can cause stress, oxygen related stresses, or hypoxia, even sometimes in severe cases. So these cannot be ignored, because in that case it can be fatal. So basically, how these, I'm not talking about bacteria, I'm talking about biofilm. So how this biofilm actually evades the immune mechanism? Very simple, because they have a covering. So this covering protects them from all the effects. For example, I have shown here in the here in the diagram, but I'll just read a few lines here so that you can correlate here. Like this biofilm matrix blocks the immune cell access. Obviously, because ultimately this you saw that only 15% of that matrix is are bacterial cells. Rest 85% is something else. So obviously that's a big blockade between the immune system and this direct contact to the bacteria. And in the same way, this is the same way that they are covered by the matrix. That's why they evade or they prevent the antibodies and complement system from their binding. They resist the they even sometimes the AMPs are what? Antimicrobial peptides, small peptides, which are being synthesized so that they can penetrate inside this matrix. But so far the studies are going on, but I cannot say that we have 100% success rate. But even these antimicrobial peptides can be resisted because of this nature of this biofilm. See, note one thing: this biofilm is a dynamic structure. It's not a static structure. So it's not necessarily that 85% is matrix only, but inside that 85%, the polysaccharide or the enzymes, their level can go up and down depending upon the bacterial species. So that was just a generalized aspect. So we kept on discussing, like they reduce phagocytosis by the neutrophils and macrophages. They suppress inflammatory immune response, or they can cause immune cell fatigue, and thus can help in development of chronic infections. and they can disrupt the signaling, not just immune signaling, any kind of signaling, which is not favorable to them. So these are a few ways, but the crux is that this covering is protecting them from all the these mechanisms which are harmful against them. Another very important question, that biofilms are also responsible for causing MDR or multiple drug resistance or the antimicrobial resistance. How? Because these matrix actually really they secrete different they secrete and they harbor different types of molecules and different types of bacterial cells. I'll just highlight here few of them. Like here you can see this yellow part here. This is basically the matrix exopolysaccharide. What it does, basically it acts as a it acts as a physical barrier, and thus it limits the penetration by antibiotics. There are several DNAs here you can see. We can call them extracellular DNA or eDNA. These extracellular DNAs actually are released by these bacteria, and they can make the bacteria which are not pathogenic them pathogenic by horizontal gene transfer. There are several reactive oxygen species released by these matrix, which can that actually disturb the bacterial cell wall, and many other parts also, but mainly they target this bacterial cell wall. So they can damage the bacterial cell wall. But what happens, this sorry, I just missed a point here. These reactive species are from outside, not from inside of the this biofilm. So what happens, these biofilms actually do not allow and to this reactive species to enter inside the matrix, and thus they neutralize them, and their effect is gone. If the bacterial cell is intact, then obviously it will keep on growing, keep on inhibiting the effects. Then there, there are quorum sensing. So what happens whenever this stress condition is there, quorum sensing starts, and you saw that quorum sensing is directly related to that changes in the cyclic di-GMP, and which can actually make them planktonic or make them sessile. Cluster them into a biofilm. Then there are several, here you can see these blue ones. These are indigo colors. These are actually viable but not culturable cells. So what does it mean? They are bacterial cells, but they are metabolically inactive, and but still they are alive. So what happens, even if this matrix is disturbed, and those antibacterial molecules enter inside it, these have capacity to actually wake up or regain their metabolic activity, and then these can take place of these bacteria and can produce those molecules, and thus fight back to those antibacterial molecules. There are some persister cells also. Persister cells are like sleeping cells. They just wake up whenever they are just like reserved armies. So they wake up whenever there is perturbation of this matrix from any exogenous molecule. There are many. There are tolerant cell, resistant cells. These are just few highlights. These are not just. This is not a comprehensive list. There are many more other things. I have just highlighted few of these. That how this matrix or when I'm saying matrix, I'm talking about biofilm. So how this biofilm actually helps in to perpetuate this MDR or drug resistance. One more thing, this biofilm can be homogeneous and heterogeneous, and most of the time it has been seen that it is heterogeneous. What does it mean? Because there are not single type bacteria. If you go here, maybe all of them bacteria are from the same species. That's why here you can see that their shape. But here I have colored them according to their characteristic. But biofilm can be polymicrobial, and most of the time it is polymicrobial. So what happens, there are different species. What happens, like here you can see we have actually classified them into two categories. I have given you here example of the periodontal disease. Even sir has also this slide in his presentation. So what happens, there are two types. The early colonizers are the primary colonizers and the late colonizers are secondary tertiary colonizers. What happens, these secondary tertiary colonizers are not able to bind the surface epithelium, but they have potential to co-aggregate with the primary colonizers. So what happens when the primary colonizer binds there, now they come and they interact with this, and this they lead to the thickness of this biofilm. Again, biofilm is not just made, I am saying biofilm, and I am using the term microbial biofilm, means most of the time I am referring to the bacterial cells. But it's not just limited to bacteria. Other organisms can also have biofilm. Like here in this study, you can see this green ones. These green ones are actually live bacteria, which have been here. The dye used for them is the cytonine dye, and then there one is F1 dye, FUN1 dye, which you can see here in the red color. They are live fungal cells. But you know that fungal cells actually they make bigger structures. I'm just forgetting the name of that structure. So here the blue colors are those fungal structure, that those here I have written fungal cell wall, but it's not just fungal cell wall. I'm just forgetting, probably we call it hyphae. I'm just forgetting the term. We call hyphae. So here you can see that a fungus, Candida albicans, hyphae, we call it them hyphae. The Candida albicans here in the blue color and the red color, and the Staphylococcus aureus, both are part of the same biofilm. So if you are giving antibiotic, you are targeting bacteria, but what about the fungus? So that's why it's not easy to treat the diseases caused by biofilm. And I'll discuss it in the upcoming slides. And here I have just listed, I will not read these organisms, but I will highlight few of the points. There are several fungal biofilms also. The protozoans can also make biofilms. Viruses cannot make biofilms, but viruses, you know that viruses can take use of anything. So they can use any living structure for their benefit. They can even use biofilm for their benefit. How? They themselves can get entrapped into the biofilm, and bacteria are there. You know that bacteriophages are actually basically viruses. So what happens? They can use them as the nutrient, and they can increase their copies. These viral particles, when protected inside the biofilm, actually can get rid of many agents which are against the bacterial biofilm, but not against them, and they can be saved. We are killing the bacterial cells, but what about the viral cells? What about the virus particles? So we can relate them as a Trojan horse. Recently you had a news that Ukraine attacked on Russia using this kind of, that actually goes into history to a Trojan horse. So what they made a horse of wooden, and actually the soldiers was inside the stomach of that, inside the belly part of that wooden horse. So similarly here you can compare that. So these biofilms actually act like Trojan horse for virus particles. So viruses do not make biofilms independently, but they can use them for their benefit. And obviously the fungal biofilm and protozoal biofilm will have slightly different kind of composition into their into their biofilm. Why it is very challenging to diagnose the infections caused by bacterial biofilm? I have listed just few of the points. I'll just quickly go. Like lack of biomarkers, because bacterial biofilms or the biofilms in general, most of the time are heterogeneous in nature. So which bacteria and which biomarkers of which bacteria we will target, because there may be other bacteria which we do not know. So it's a very difficult because of the lack of universal biomarkers. Most of the time we take directly from the sample. So most of the time they also give false negative, because because we do not have any negative culture against them, so as to compare them. And whenever we use any imaging technique, for example, I have just listed here that CT scan or X-rays, they cannot distinguish these, they cannot actually give any information about these biofilms. And lab conditions actually most of the time fail to replicate the natural biofilm. Nowadays, many techniques are developing called 3D biofilm printing, but we don't know. They are just in their incipient phase. And similarly, we can say that biofilm or implants often require removal of the diagnosis. So diagnosis itself is a big problem here, because if you suppose you got the symptoms related to a particular bacterial species, but if it is a heterogeneous, then what about other bacteria? You are killing them, and if you are not using, suppose, a broad spectrum antibiotic, then they may get safely escape these treatment. There are many other things, like there are less diagnostic tools, like I discussed with this confocal microscope or this electron microscope. So obviously they are very costly and they cannot be set up into a pathology lab. I'll just go to section 3 and section 4 very quickly. Therapeutic studies, I have just included 2-3 slides here, not much. So I have listed here all the traditional and the new emerging techniques to combat these biofilms. I'll start with here natural products. Natural products, you know that there are many, most of the plants that here, indigenous plants, mostly we have seen like neem, tulsi, all of them have simultaneously antifungal, antibacterial, anti-inflammatory, antipyretic, many kind of properties. So these natural remedies can act better than antibiotic, because they are targeting this heterogeneity of the biofilm also. And but again, without diagnosis and without keeping a track of them, we cannot just rely on them. So we need other methods also. Traditional antibiotics have several limitations. One of them big is the MDR, multiple drug resistance. So we cannot only rely on antibiotics. And you know that now this is the era of synthetic antibiotics. So we are repurposing or reinventing the different kind, the same antibiotic for different bacterial species. But bacteria have smaller genomes, so they are developed. They are also copying up and copying up these treatments, and they are developing further resistance. And those secondary or tertiary resistance that they are developing are very hard to treat. Nowadays, there is a technique called phage therapy. Phage therapy, I know one of the professor who is working in the phage therapy. What they are doing here, they are infecting those biofilms with the help of certain viruses. And these viruses, you know that they can kill and use that bacterial cells for their nutrition. So here this technique can be useful, but again problem is that we have to deal with the viruses, because viruses again have very high rate of mutation. Then one of the strategy is here to stop this quorum sensing. And if the quorum sensing or the communication between bacterial cells are stopped, then obviously they will not be able to form this biofilm. If there is no biofilm, then their effects will be also very less or no effect. And nowadays monoclonal antibodies are also being used. But again, monoclonal antibodies versus polyclonal antibody, it itself is a big debate here. So I'll not go into the detail. Here you can see these when these combinatorial, we can use them in a combinatorial method manner, or I mean in the combination. And whatever happens, these multi-target strategies actually can help us to get rid of these bacterial infections. But we should always be prepared for the mutations and the drug resistance developing very quickly. Now here comes a saying that loha loha ko kaatta hai, so we can cut iron through iron. What if we use bacteria against bacteria? So there will be many problems which will be solved. Like one of the biggest problem is the multiple drug resistance. What happens when the bacteria, these probiotic bacteria are class of bacteria which we can call them beneficial bacteria. They do not cause disease. They are like most of the lactobacillus species. They in fact are required for good health. What if we study them and we find out new probiotics, just like we found out new antibiotics? So just like there is an era of new antibiotics, can't we have an era of new probiotics? I mean, the bacterial species which are naturally not probiotic, they are also not pathogenic, and we can do some genetic modification and enhance their capacity to bind to the same receptors where the pathogenic cells are binding. So what will happen? There will be a competition between those cells. So suppose somebody gets infected from a particular pathogenic bacteria, and we have some technique, we identify what those bacterial cells can be and what those organs or body organs can be, and we have a specialized medicine, a specialized probiotic, and we give them. It will go there, it will compete them, and we can also give them prebiotic. Prebiotic, what is prebiotic? Prebiotic are those food materials which can help the probiotic to grow faster as compared to the pathogens. So if all those all those receptors will be occupied by these probiotics, then obviously body will flush out those pathogenic cells, and we will get cured in cheaper manner, less threat of mutation. See, threat of mutation is still there because horizontal transfer of gene is there, but right now that need to be studied. But we can get rid up to a certain extent from this drug resistance. Section 4, actually I wanted to cover it in detail, but right now I have just put a summary. So right now there are several innovations going to combat these bacterial biofilms. I have listed few of the chemical methods, some physical methods, and some biological methods. You are familiar with the biological methods, so I'll just take name of few one, like bacteriophage, those viruses against those bacteria. Anti-biofilm peptides, which we can call anti-biofilm peptides, AMPs, those small peptides which can actually penetrate inside this matrix. There are many natural products. So we should go back to nature, because the natural components have not just unidirectional therapeutic use, they can provide multidirectional therapeutic ability. Many enzymes, biosurfactants, many more things. Now certain things like if we talk about not biotic but abiotic surfaces, like dress, like PP kits, like table or anything, then we can use ultrasonic treatment, ultraviolet radiation, ionizing radiation to disturb those biofilms. There are certain chemicals that we can use through different methods. Actually, I'm not going into the detail mechanism, so I have to skip. But I would like to highlight two of them. One is the smart surface coating with antimicrobial biomaterials. So new bioengineering is happening right now. This I found from a recent review paper that there are certain chemicals which here you can see this metal nanoparticles, titanium oxide, here antifungal, antibacterial, antiviral coatings, different anti-biofouling like this chelating agents basically. If I talk about chelating agents, deuterions, polyethylene glycols, these are the AMPs, and some other there is one thing called chitosan. Chitosan are actually natural polysaccharides with broad spectrum antimicrobial activity. So what is the idea? We synthesize them in the lab and we coat the medical devices. So medical devices will themselves have an antibacterial property. Just like most of the common is like the PP kit, wound dressing, orthopedic, basically all kind of implants. We can use them. So these are also a new area of research right now. And I skipped all, but I will also talk about this artificial intelligence and machine learning here also, because this is totally new, and I maybe those who are not familiar with artificial intelligence and machine learning, actually this can solve many big problems that are not solved till now. Suppose we have a database of all the biofilms from all over the world, and we train a large language model to identify, to analyze that bacterial biofilm. So it will be just like clicking a photo of the bacterial biofilm, uploading to a server, and server will give you answers that this biofilm, it belongs to this particular bacterial cells, and now you have therapy against those bacterial cells. So here computer or computer can help us. There can be a species identification. If we train the large language models or the artificial intelligence using different kind of electron micrographs or different kind of different kind of biomarkers, we have also now another field is going on where they are trying to make biosensors for biofilm. So if the data of those biosensors are fed it up to a database, and we just simply upload our diagnostics there, and it will give us, this is the bacteria, these antibacterial strains should be used. This is I'm just skipping here. I'll not go into detail. The idea is that making a database of all those images taken from the confocal microscopy or electron micrograph can also actually enable artificial intelligence to identify and simultaneously suggest the cure or treatment against this. So I'll stop here. If you have any questions regarding this, then otherwise I'll just quickly go to second part if you're interested, because I just want to go that part only if you're interested. That's a structural biology part. I'll just give you a this is my own work. I'll just quickly skip few of the slides. Okay, as I told you, our lab is actually divided into two parts. Some of the students, PhD students, are working on the pathogenic strains, and some are working on the probiotic strains. Yeah, quickly, I'll just take 10 minutes only, because I will not go into details. I'll just give you idea that how structural biology works. So what happens, we were working on a particular class of pilus. We call it shortage mediated pilus assembly. So what happens, as I told you, there are 3 subunits. So these the genes for all these 3 subunits are clustered into a single operon along with an enzyme called C-type sortase. The C-type sortase actually assembles these subunits into a longer pilus, and they, somewhere in the genome, not inside this operon, there is another enzyme called sortase A enzyme, which we call housekeeping sortase. This housekeeping sortase actually anchors this fully mature pilus on the polypeptide on this bacterial cell wall. I'll just not go into this what is YPK and what is LPXTG, as sir has already discussed. So okay, this is interesting here. So you can see that there are many diseases like animal blotting, this abdominal blotting, acid reflux. These are normal diarrhea, infectious disease. Even exercise induced immune suppression, and this depression. Here you can see the inflammation, and here this focus here in the depression also. If you are having good probiotics, it can actually make your mood also, and it can give you better memory and less anxiety. So why not to include them in your food? So just a quick liner here. So this is how I told you that probiotic bacteria actually can compete with this. Green ones are probiotics, red ones are the pathogens, compete with them for the same receptors, and this is how they can nullify the ill effects of the pathogenic bacteria. Different studies where So basically why we study the pilus polymerization in case of probiotic bacteria, because you may ask me a question. We study this pilus formation in case of pathogenic bacteria because they are causing disease, so we need cure. But probiotic bacteria is not causing anything, then why we are interested in them? Because the biggest answer is that they are opening new technologies, which I am not covering here, but if you, I would encourage you to go and Google out. So these are like I have seen here, I have not listed here, but there are many techniques, technologies developing throughout this, using the structural information from these probiotic bacteria. The bacteria that I worked during my PhD studies was the Lactobacillus ruminis ATCC 25644. So this particular strain is actually from human gut. So humans also have Lactobacillus ruminis. Here you can see the presence of the pilus on its surface, and in the genome we found that it has that cluster of the operon containing the genes for all the 3 subunits, along with the sortase enzyme responsible for the pilus polymerization. Here you can see an electron micrograph where the antibodies against those subunits have been used to identify them. And here are several studies where it was found that if we give this Lactobacillus ruminis, okay, this pilus of the Lactobacillus ruminis is named as LRP CBA pilus. What is LRP? Lactobacillus ruminis. Now it has been renamed as Ligilactobacillus ruminis pilus proteins CBA. What is CBA actually? Here, this is the C, this is A, this is B. So when I am saying A, B, C, I am saying A for shaft pillin, B for basal pillin, C for tip pillin, because it's the name of the gene which are encoding those proteins. So we had 2 questions. One question was that as you saw when sir was showing that the basal pillin was absent there. So a question was that here is the basal pillin, and it had several, it has these subunits had actually very low sequence identity to other known pillins. So it immediately hinted that the structure of these pillins could be totally different. If the structures are different, then maybe the mechanism is also different. And we found that yes, mechanism has slightly difference as compared to the previously known. So first objective was to understand how these are polymerizing, and the second objective was that interestingly it's found in the intestine, but it does not have any mucus binding property, and it is autochthonous. That means autochthonous means it is present inside our gut by birth. So we had to study that then what are the other ECM proteins. So I will just focus here only on this part very quickly. So we purified that shaft pillin. This one is the shaft pillin, purified, crystallized, and we had very high resolution data. And I will not go into technical details. This protein had actually 3 domains, but we were not able to solve those structures, because for solving we needed some level of sequence identity to other proteins. More than 25% or at least more than 40% will be preferred. But this had very less sequence identity. So we used a novel method. What we did, we truncated the top domain, the N-terminal domain of this protein, and then we crystallized it. Then we collected data for that also. Okay, I'll skip these things because these are highly technical. So it may be difficult for some people to understand here. So what we found that from our crystal structure that we solved, we found that, so this is one subunit. You can see this is the N-terminal domain, middle domain, and C-terminal domain of the shaft pillin subunit. So when we docked them, and we found that the tail of the first molecule is sitting somewhere and making some interaction that sir has also described, similar kind of interaction. But when we tried to make a polymer, then we found that actually it is a zigzag pilus. And interestingly, So far, never ever we have seen any zigzag pilus in bacteria. Some fungus, in case of some fungus we had seen, but this was the first time that we are seeing here. So we were actually that time little not, we did not had confidence whether we have correct data or not. So what we did, when we went back to the crystallization condition, and we found that this crystal grew at acidic pH around 4. So what we did, we tried to crystallize it into normal physiological pH, but we could not get any success. But we got another confirmation of the same protein again in some acidic condition, and here you can see this N terminal is slightly more shifted. If you see clearly here, slightly more shifted, and when we created a polymer, this polymer was slightly different from that. So what we did, when we tried many times, but we could not get a crystal at physiological pH. So we used AlphaFold model, and we modeled this structure using the sequence, and the AlphaFold model gave a linear sequence. This linear sequence is the conformation mostly found in the pilus structure. So what we did, when we created a polymer out of it, this polymer resembled to the most common piluses ever seen previously. So when we superimpose these three structures, what we see, this middle domain and C-terminal domain are exactly similar. Obviously tail is free to move, so tail will not superimpose. But here in that N-terminal, you can see there is remarkable difference. So we tried to find out now, we had a question. Now which one is correct one? Which one is present in the nature? That bent one or the linear one? Because we got the crystal structure of the bent structure. AlphaFold says that it should be linear, and the linear model actually matches to what we have been, what the other people have solved so far. So now we had a question that which conformation actually exists in the nature. So there is another technique called small angle X-ray scattering technique. Very few people are expert of it. So I learned it, I solved it. I will not go into the detailed technical part. So what happens when we grow a crystal, a protein, and we put that crystal in the in front of the X-ray beam, that crystal gets diffracted. We get diffraction images, and then we solve the structure. But in this technique, what we do not grow crystals. What we do, we take the protein in solution, and we directly put in in front of the X-ray beam. See, the X-ray beam will not give any diffraction pattern now, because it is not a crystal structure. But what happens, it gives very weak, very low resolution envelope. So this envelope actually helps us. So what we did, we purified the protein in the physiological pH and collected the data using the SAX, the short form is SAX analysis. So we used SAX analysis, and what we found that the SAX analysis says that the protein structure must be linear. So though we could not get any crystal structure for any other conformation apart from bent one, but now we had proof that in the physiological pH, actually the structure should be linear, not bent one. So that means then our crystal structure that we got was hinting something else, never seen before. So what we did, we then tried to measure this movement, the angle of this movement, and when we measured them and listed out, then we found that there is certain degree of rotation, certain degree of translation, and certain degree of closure. So we gave, use different techniques. Here you can see this. Here I have just skipped one important part. So if we go to the N-terminal part of this structure in the crystal structure, so there is a particular loop that we call AB loop. So what is AB loop? The loop which, you know that protein structures have three components: alpha helix, beta sheets, and loops. So this particular loop was between the beta sheet A and beta sheet B. That was absent in the crystal structure. And this is where we got hint that because this is getting disordered, because disordered structures cannot crystallize. and probably this is why the crystal structure is giving a bent structure. So what we did, we modeled that missing part, added back to the crystal structure, and then we performed docking, bent to linear, linear to linear models, and we found that here you can see that this is the C domain of the one subunit and the N terminal domain of the other unit, and I have highlighted here, you can see the tail is sitting properly here, and this green one is that AB loop that we actually did not get in the crystal structure, but we modeled. So what we got here, that there is a particular linking lysine in this AB loop, which was actually shifted away when this AB loop was disordered. But when this when this AB loop was added back, then this particular lysine link, which we call linking lysine, because this actually links this one subunit with the other subunit with the help of a isopeptide interdomain isopeptide bond. So you have heard about peptide bond. So I'll encourage you, go and read what is isopeptide bond. So I because of lack of time, I will not describe here. So there was an interdomain isopeptide bond or intermolecular isopeptide bond, and we got this structure. This is the 2D diagram here. You can clearly see this one, the threonine of the this tail from the first structure, and this linking lysine you can see here. They are in the close vicinity. So what we did, we just tried to find out proof that this conformational change is actually happening or not. So what we did, we purified the protein again in the acidic pH and in the physiological pH, and we went for we went for this fluorescence studies. This is called the intrinsic fluorescence based on the intrinsic tryptophan. So what happens if there is a tryptophan? Yeah, if there is a tryptophan, what happens? It absorbs, and if the tryptophan is open to the solution, it will absorb, and if it is hidden, it will not absorb. So the molecule, when it is bent, so it is open, so it will give different reading, and when it is actually in the linear molecule, it will be hidden, buried, so it will give a different reading. So here what we found, that at the pH 4.8 the absorption is high in the fluorescence study, but at physiological pH it is low. High in case of acidic pH means what? That intrinsic tryptophan, which is at the junction of this N-terminal domain and the middle terminal domain, is actually getting exposed because the N-terminal domain is bent. So it gave clear hint that at acidic pH it will be bent, and at physiological pH it will be linear, even in the natural condition also. So we again validated it using another technique that we call here dynamic light scattering. So similar kind of a study validated it. So we compared it to the previously known other proteins. So I'll just skip. So ultimately what we, I have skipped few points here. So we, the mechanism we actually discovered for the first time, we gave it a name, unique closure and twist motion of motion for the assembling dynamic pili in L. ruminis. This is my last slide, so I'll just take 1 minute more. So what is the summary? So what happens? First of all, the tip pilin, which is LRPC, actually gets secreted to the membrane, to the cell wall, and in the membrane there is this housekeeping sortase. It interacts with this housekeeping sortase, so housekeeping sortase actually grabs it, and then similarly a nearby another sortase, because sortase are scattered inside the inside that membrane. So another sortase similarly binds with the shaft pilin, the backbone pilin, and what happens? This backbone pilin goes a change. You can see here the N-terminal change, and then when this linking lysine is exposed, it attacks to this bond between the active site cysteine and the threonine orbitopilin and breaks it, and this gets loaded on the top of it using that threonine lysine inter-domain or intermolecular isopeptide bond, and this keeps on, and this pilus keeps on growing. So what decides the length of this pilus? There is no set rule. What happens whenever actually I have stopped here, but there are a few more things I will not I have not shown here. I have just shown here that how the pilus polymerizes. But if you remember sir's slide, what happens whenever basal pillins comes? So basal pillin is basal pillin, which is at the bottom, does not interact with this sortase, this housekeeping, this C type sortase C. That interacts with that another sortase, which was not part of the operon, that is housekeeping sortase, and then this whole mature pilus gets shifted on that shifted to the housekeeping sortase from this pilus specific sortase, because then just like this attacked here, the basal pillin attacks here, and the whole assembly gets fixed with the help of that enzyme into the polysaccharide polypeptide part of the cell wall. This is how the bacteria grows. So what one line message, take home message is here. Bacteria, this L ruminis, okay, I am done with this one. So L ruminis, whenever L ruminis bacteria is actually autochthonous. Autochthonous means it comes inside our body during birth. So it's present in our intestine from birth. But if we are saying that we can give it as a probiotic, so if you are taking it as a probiotic, obviously you will take it from oral route. It will go into stomach, and there what is the pH level? Highly acidic. So how this bacteria is surviving when taken orally as a probiotic into that harsh acidic environment, and how it is escaping that or surviving that and going to the physiological pH, it's somewhere 6.8 inside the intestine, and there that is the actual niche of this one. Now we had answer. What happens? The bacterial pilus actually, when it enters into the stomach, so in the acidic environment, what happens? This pilus actually forms this bent kind of a structure. So it becomes a spring-like structure. So it reduces the length of its pilus, and it does not interact to any receptor. But when it goes to the intestinal physiological pH or the slightly alkaline pH, then what happens there? It gains the linear conformation. Now it was like this, now it gains like this, and it grabs the receptor, and that is how it binds there and grows into a biofilm. Okay, thank you. Sorry, I took a lot of time. If you have any question, you can ask. नहीं अभी
telling us you are sitting And then the second time they said, whoa, I know this enemy, and I know I can kill this. Then what about next time? So their response always like a increased every time. Like the innate system, they're kind of like pretty constant, right? They're kind of boring, but they are okay. But the adaptive system is more inspiring. That means they can recognize the enemy, and when they come back, they see that second time, they will attack violently. So they will precisely kill that organism. So this is the layman's language I'm using to explain that how adaptive system works. Got it? And the active molecules are cytokines, the protein molecules or secreted molecules, and then the antibody. These are the two. And then the cytokines work by using specific cell type, right? So if macrophage is the first responder trying to kill the organism, and I mentioned a little bit about the trained immunity, right? They can be trained, but the adaptive, essentially they could be more, I mean they are robust. Usually pathogens may not be able to destroy it. So they are same person, but when they are trained or when they are part of the adaptive system, they work much vigorously. Okay? And they are also again precise. They will find a way to control it. And so all of those things there. And then the other part is that adaptive, we try to break it down into two systems. One is called active and another is called passive. Active means if you inject an antibody development, but we need something pretty quickly to protect you. So for example, active system would be, you heard about tetanus, right? And you go to the doctor, they will give you a tetanus shot or vaccine, right? And then you take it again after some time, right? Do you know what is the time window? Say 6 months, you said? Something like that, 6 months is the time window. Anybody knows that how long the protection lasts? How long before the doctor might ask you, you have to take the tetanus again? Anywhere between 8 to 10 years. That's the long, that's the duration. So that means that long is the memory function, right? Okay, so whenever we say the vaccine has a certain duration, and that's the length of the memory response. Everybody clear on this? Are you clear on what I'm saying? If I say the vaccine lasts for 14 years, right? So usually these memory cells are highly, or they could be active or can be activated within the time window. After that they may die, they slow down, or they may not respond any. That's the time window we present. So we talked about pox, right? Smallpox. What did we talk about? Or chicken pox. If we have it, protection for life. What does it mean? That means lifelong protection means your memory cells against this pox, they live longer, right? Some vaccine is only 1 year or less 2 years, right? Or short duration, because their memory response is that short time. If you get infection after that, you may get sick again. Okay? So that's the secondary immune response. And when you say passive immunity means, so for example, you are walking or hiking or walking in the field, suddenly you're bitten by a snake, right? You need protection against snake bite. So do you wait for active immunity to happen, or you need something immediately, right? So usually we give anti-venom antibody. So that means that antibody already being made in different animals. For usually horse they use to make that antibody, and we keep them in the vial in the hospital. As soon as you get there, if you say that I got bitten by cobra, they have a vaccine against cobra antigen or cobra toxin, and that's actually you are receiving antibody. Everybody clear? You are receiving antibody against the cobra toxin, and that's called passive immunity. It's not active because you don't have time to build your immune system to make that antibody. But in this case, you are using just the serum harvested from animals which already received that venom. Any other example you think of? So there are few of them I wrote there. Also botulinum toxin. Botulinum toxin also works that way. So if you eat some food, you have botulinum toxin, Clostridium botulinum, and that toxin can cause paralysis. People can die. Okay, it's the same thing. So when you go to hospital, if you doctor decide that you have a botulism, and they will give you anti-botulinum serum. Again, they have that, and because it attacks nerve, so you need to go as quickly as possible. Same way as the snake venom. So they will give you anti-botulinum serum. These are real life story I'm telling you. So it's not just education purposes, also for protecting against, if you suspect that you ate certain food, likely had a botulinum toxin, and then you need to go to hospital as quickly as possible. Usually they all carry this anti-botulinum serum. I'm not really sure in the US, excuse me, India, but US, like any hospital you go, they will have that one. Because it's so serious. It's a matter of time. Why? Because the toxin binds to nerve, okay, nerve receptor. So sooner you intervene, faster result you can get. That's how it works. Snake venom also same way, okay, so that it antibody neutralize the toxin. Okay, so these are example of active and passive. You will everything remember. Okay, so when you look at the also immune system, these are some of the specificity I already mentioned. Okay, you can read this, that way you learn it. And so one of the characteristics of the immune system is that always increased. If this is a primary response, the day you first got exposed to the pathogen or toxin, or if you taken a vaccine, right, that's the primary day, the day one. So you get a primary response, very mild, weak response within say 3-4 days to 7 days. But then if you take a booster, we all heard about booster, right, and then the response would be significantly higher. This is always the characteristics of adaptive immune response. Okay, if you don't use this immune system because you don't need it, if there is no pathogen coming in, essentially my immune cell say, oh, let's stop working. This is how the immune system goes away, okay, or become weak. This is how the system works. Everybody got it? Because there is no reason to stay active all the time if there is no threat. So that's the time we have memory cells. So before I get there, I want to tell you, yeah, I'm there. But one thing I want to mention also, two types of response we get. One is cellular immunity. One is called humoral immunity, right? Cellular means cells are involved in taking care of the pathogen. Everybody clear? Cellular is the cells, immune cells. And humoral, where is that antibody or secreted molecules are responsible for taking care of the pathogen. Okay, these are the two types. So as I said before, how do we get this response? How does it start? Think about it. Okay, the whole immune response. Let's look at this way. So all of those immune cells sitting in the body. So if you go about stem cell, we talked about this morning, right? We have bone marrow stem cells. Also we have stem cells in other primary lymphoid tissues. But bone marrow is the primary site a lot of our immune cells come from. So the first thing you ask that question, how do I get activated? How do I know that which antigen to respond to? All of you are standing there in one location, right? The antigen come in, and then what happens, you remember that each antigen has a choice for one specific cell type, whether it's a T cell or B cell. Everybody clear? So that's how they become specific. So for example, if you have A, B, C, D, these are examples of clones or the cells, whether it's a T cell or B cell. Try to follow me. Now once the antigen comes in, this antigen can activate one of these clones. Okay, let's say example A, this antigen came in, well, it's bound to the C, let's say it is C. It bound there, right? Now the clone C try to enhance their population. So what happens is that C binds to an specific antigen. So if it is a virus or bacteria or their proteins or toxins, whatever may be, as binds, then this specific clone get activated. That's the first step. And then it starts actually, once it is activated, then it starts to proliferate. It divides. But this one recognizes this antigen. And then once it is proliferated, they call differentiation. Means it goes through a step where they will produce antibody, right? In this case, this is Y shape, the antibody, or it could be cytokine, and then one population would convert into memory cell. And as I mentioned last time, the memory cells are like a night guard, right? They call body surveillance. They are surveilling your body continuously. What they are looking for, any kind of antigen or pathogens coming in. As soon as they come in, they will what? They will play the whistle, right? Remember last time? Or they will try to differentiate. They become, think our memory cell goes back here, they will proliferate, they will differentiate, and they will do the same thing. So this is how the story works. Whether it's a cellular or humoral, depending on what molecule. In this case, I am using example of antibody. So it's a B lymphocyte, and this could be, say, cytokines, certain specific cytokines. Okay, the small molecules. That could be T cells as well. So you have a T cell memory, also B cell memory. Okay. All right. So the whole story is that where they come from. I think the primary I already mentioned. They're like elementary school I mentioned, right? You were born, you go to elementary school, you learn about it first. And then once you learn about it, and then you know how to defend it. So the lymphoid tissue, I mentioned spleen, lymphoid tissues, and these are some of those organs. So this is just showing where the lymphoid tissues are in the body, right? And mouse, we do regularly experiment with mice. I just want to show you that mice has similar things. So they have thymus, and they have the spleen here, and the cecum colon, and I mentioned about the Peyer's patch. If you look closely, you may be able to find the Peyer's patch. And those are the lymphoid tissue, where the T cells, B cells, also the antigen presenting cells, they are all localized. Right? So you need 3 units. One, going to get the antigen, bacteria come in, they process it, they cook it, they offer it to the guest, in this case T helper cell. We ask the helper cells, they will produce cytokine, they will activate B cell, and they will make antibody. This is the story. If you remember this, you will never forget how immune system works, at least from the adaptive side. Very simple. Antigen presenting cells, which are macrophage or dendritic cells, they present the antigen to the helper cell, they will produce cytokine, and that cytokine help B cells to produce certain type of antibody. Okay? Or T cells, the T helper cell, they can also ask other T cells to help. And that's how the cooperatively they work. And that's how the adaptive immune system works. That's the more simplified way to explain that. So primary lymphoid, again, you can read this. You are going to have these slides to find out what are those organs, how it works. If I have a lot of time, entire semester, I'll teach you every detail of it. You know, this is how it works. But right now I'm trying to give you the big picture, how things work. You already saw that gut, right? So if the pathogens coming in, this is dendritic cells, this is macrophage, they will take that antigen. The way it works, actually, the antigen presenting cell, I told you about the cooking, right? Cooking part. And literally they cook it. What they do, if you take the big raw product, right? And you cannot eat the raw stuff, right? Like that. You have to cook it. So what they do, actually, once they get the bacteria inside, they will cut it into small pieces, chop it into small pieces. And only a certain fragment of that they will present to the T cell. Only a small fragment. What is that fragment? What can you tell me about that little fragment they present to T cell? What is unique about that? What is specific about that? Anybody? So now imagine the picture. So the whole bacteria, right, big cell, the macrophage grabbed it. And then bacteria has a lot of components. We learned that, right? the cell wall, the fimbriae, the flagella, they produce a lot of proteins, right? A lot of stuff. They got the hundreds and hundreds and hundreds of different types of proteins and molecules. So tell me, when they cook it, what part of that they are going to present it? Do you think they are going to present all those hundreds and hundreds of molecules, or they are picking certain molecules to present it to the guest? Something, oh yeah, somebody said that antigenic molecule or immunogenic molecule. Okay, what it is actually, this macrophage, this dendritic cell decides what am I going to present to T cell, so the T cell would be highly active. It's like your Valentine's Day. What do you present? Right? Flower, chocolate. Why do you do that? You want to make your partner happy, right? So that you remember. But you are not going to present something like a kulfi or regular sweet or whatever or poha, right? I mean, those are not something special, right? You want to present something very special, and that special is antigenic or immunogenic. Not everything is good in that mass, right? Only few things are small, and that little thing that's highly special. Like your cake today, right? You enjoy the cake, right? So we didn't present everything, the box, right? Or the spoon or the candle if you had it. No, there is a little thing, right? That's unique. So what happens, literally, they break it down to small pieces and they find certain amino acid sequence which is highly unique and antigenic. And that's what the T cell grabs it. And how the presentation works, I don't know whether I'll be able to get to that. The way it's like, how do you present when you go to temple? How do you present it? Tell me. Or even if you present somebody, do you throw it away? Hey, there's a temple god sitting there, or your guest is there, right? You want to give them, serve them. How do you serve them? Really nice, decorated, right? With flowers, with prayer. So if you look at these cells, this is true story. I'm not making it up. What is this? You go to temple. How do you offer? Would you offer like this, right? Have you seen this? We offer this. So this macrophage or dendritic cells, literally, they have an antigen presenting molecule that look like this hand. And so the hand they present, it has a certain size. Okay? It can hold only certain size molecule. And that molecule, anybody guess how big are those? In the molecular level. So most of the time they present protein antigen. That's a clue. Okay? In the protein antigen, you break it down, you get what? Peptide bond, and then you see the backbone is amino acids, right? So there is a limit. So when they offer about 10 to 12 amino acid long small fragments. Everybody clear? And literally, if you look at the electron microscope picture, it looks like a hand. So they just fill this gap, and so that's called major histocompatibility complex, called MHC. That's what they use to offer to the T cell, and this T cell can recognize only when presented with that, like this, offering truly, and that's when they respond to it. And so this is so fascinating. They are not going to recognize it until they are properly presented, and that unique amino acids are actually antigenic. You mentioned that they are special, right? And that specialty is that they are antigenic. So you do break it down into a lot of pieces. Not all structure are going to be antigenic. Are you seeing this? So they may have a unique amino acid sequence, unique structure, okay, unique folding. Because you saw a lot of those protein structures that people talked about today, and that's what makes it unique. And this T helper cell, well, it recognizes it, it produces some cytokine, and it can tell the B cell, oh, I need to produce antibody against tetanus toxin or botulinum toxin, right? And then you need to know what type of B cell, what type of antibody they are going to make. You know that antibodies are antibodies, right? But if I tell you that there are different classes of immunoglobulins, right? IgG, IgM, IgE, IgA, those who have learned immunology, those classes depends on who actually receiving and what type of molecules they are secreting. That's how you get different isotypes of antibodies. So all starts from presentation. You see this? Because they cook, they need to be a good cook actually. They need to process it well. They need to know how to do it. And that's what's so fascinating about antigenicity we talked about, right? Then also may contribute to the memory response also. And how effectively these antibodies or immune system works for you to help protect against diseases. Are you seeing this? I literally covered the entire adaptive immune system, and that may be many pages long. Everyone understood the story now? So if I to show you, let's take a, I'll get to that. I think let's see if I have that antigen presentation. Went so far. Here is an example. You see this one? This is the dendritic cells, and this is MHC molecules, and that binds the antigen, and this is presented here. I don't know if I have another one. Okay, best place to show you is right here. So antigen presentation. This is macrophage. It gets this antigen. See this cup or the presentation, and then this T cell recognizes it. And as soon as it activates, then it's producing cytokine. Okay, and that's helping the cell-mediated immunity. But also that cytokine is with Th2. It's a Th2 type, helper type, and produces cytokine. Then now B cell going to produce antibody. And what type of antibody they are going to make? Let's see if I'm jumping quite a bit, just to show you how this works. The whole story I told you. Because of the limited time, I don't have that right now. I think I have to go further down that way. So this is also another way to present it. Okay, this is the basis of understanding immune system, how immune response works. The who is presenting also makes a difference. So this is macrophage, dendritic cells. The B cells also present. They are also notorious for presenting a good type of cells. So this cell type. So these are the two types of. We mentioned of the stem cell. I'm going back a little bit, and then here is the T cells. This is the T cell. This is the B cell. This is plasma cell, and these are the Th1, Th2. All those different types of subcategory of T cells. Those who have taken immunology. And each of them has a different function. And this side, on the left side, when you go to see a doctor, they do a blood count, right? They do look at the type of blood cells you have. Whether it's a neutrophil, monocyte, basophil, eosinophil, all of them are indication of certain type of diseases. Okay? So for example, you are suffering from allergic diseases. What type of cell you will see? Basophil cells, right? If you are suffering from parasitic infection, eosinophil, right? That count would go high. If you are suffering from bacterial infection, neutrophil count would go up. The viral infection also you will see a lot of neutrophil up. And so whenever you look at this cell population on the left side, right, and that's the indication of that you have type of diseases you have. Everybody clear on this? How this actual the cell type and who respond to what antigen. So I wanted to tell you that antigen antibody presentation and dictation. And then other thing also may be critical for your understanding of immune response is that what classes of immunoglobulin you are going to produce, right? And so let's go to the B cell. Again, I want you to read this. There's a lot of information there in that, and I know with the interest of time we will not be able to cover all of this. So essentially there are a lot of whole immunology there, right? So antibody structure I don't need to explain, right? Probably you have seen this. This is how where that antigen binds, and this is the structure, how it look like. This has different parts and different function. But what I want to tell you is that, let's see if I have that. Oh, so when you, as I mentioned that B cells are going to produce antibody, and each type of antibody also respond to depending on what type of disease you have. Okay. So, for example, when you take vaccine, right, practical example, and if I test your blood within like a 4 days or 5 days, what type of immunoglobulin I'm going to see in your blood, okay, versus if I test your blood after like a 2 weeks or 3 weeks later, what type of immunoglobulin I'm going to see, okay. Or let's talk about vaccine. If I take your blood, I want to test it, see if you are suffering from allergic diseases, or hypersensitivity, or parasitic diseases. What type of immunoglobulin we're going to see? Because immunoglobulin also, like the cell type I mentioned, immunoglobulin also vary between patients or type of disease you have. Okay, when you saw this immune response, right? So when you look at the immunoglobulin, we have 4 different types of immunoglobulin. One is called IgM, IgG, IgA, and IgE. And so if you look at that, I didn't go through these structures. So this is IgM structure. This is kind of like a 5 immunoglobulin, you attach them together. This is IgG structure. And this is IgE. Which diseases you see IgE? Allergic, right? Perfect. So allergic diseases you see IgE level in high. And IgA, where do you find IgA? Mucus. I told you this morning, right? The mucus has IgA, and blood serum has very little IgA. But they are usually mucosal immunity, IgA play a role. And this is called IgY. What is IgY? Anybody heard about IgY? No, none of you? Huh? At least one of you should know. What is IgY? That's where I have fun. Yeah, usually this is unusual, right? Anybody? You heard about chicken, egg, yolk, Y. So IgY is a chicken IgG equivalent. That's why I call IgY. Okay. Very rarely we talk about IgY because it's a chicken, right? We eat egg. You can have IgY in yolk, egg yolk. Okay. So this is a good way to do this. If you inject a chicken an antigen, you can harvest antibody from egg. You don't have to get it from the blood circulation. Think about it. This is a lot of now new development. It's not a new technology. It's old. And what we are trying to do, a lot of times people are trying to do is that, what about, well, I mean, if you are vegetarian, of course, if you are avoiding egg, probably we cannot use that for you. But a lot of times what can be done is that you can introduce certain antigen into chicken and harvest the egg, and you eat the egg for as a treatment, therapy, because you got this immunoglobulin there. Okay, the structurally little different, but this is that. The beauty is that all the antibodies are actually in the yolk, and so you purify from the yolk. Unless blood circulation or serum, it will be hard. So this is what the IgY. So what I was trying to tell you is that when you look at in the first phase of the immunization, you usually see IgM. Okay, IgM is very unstable, and it's not the desirable antibody. But the beauty of IgM is that it got 5 arms, like Macaulay. They got number of arms, right? But each arm has a lot of function, right? Each arm can do, and essential IgM does that. But the thing is, it's fall apart. It's so unstable structure. What we want for our purpose, we want IgG. IgG is the most desirable, stable immunoglobulin. So how do you get to that? And why it converts? So most of the cells would be producing IgM no matter what in the beginning, but they convert, they switch. What made them switch? You seeing my logic, what I'm saying? In the beginning, all of you, learn certain things, specific things. But then eventually you specialize in certain function. Okay. So here, what make them produce these different immunoglobulins? So the beauty is very simple. So if you take this clone, remember this T helper cell, right? The type of antigen came in, and they will ask the resting B cell to get activated, and then eventually they are going to divide into different type of immunoglobulin. So if you have been exposed to a lot of allergen, say pollen or any kind of small molecule which you are allergic to, so what you will do actually, this allergen can activate certain type of cytokine, and then this cell become IgE producer. We started all of them. Initially they will be making all IgM in this phase, but as soon as which subset of T cell become activated, they are going to produce different types of cytokine, which is the juice. It's kind of activate them. It's like an energy drink, right? You take energy. Are you familiar with the energy drink? Yeah, I mean it's like a lot of calorie, a lot of caffeine. It will wake you up, right? And so depending on what are those, these are the specific cytokines produced by this T cell, and then depending on what cytokines you have, then going to make this antibody, right? Why do you need to know this knowledge? Let's talk about disease. Do you like the allergic disease? No, right? It can kill people. Somebody allergic to peanut, I mean, there was a kid actually, he called his parents that, oh, I did drink some juice with or some kind of beverage, and then within 1 hour this person died. The sad story is that the allergic diseases, when it is severe, it can kill people instantly, almost. Okay, so a lot of times the treatment would be, what do you do? What do we learn from here? See this one, IL-4. So what is that? IL-4 is the cytokine. If you have anti-IL-4 antibody, it can neutralize it. So there will be less IgE production. So number of ways you can treat it. But allergic diseases are so, almost there is no treatment except you take antihistamine, right? Because you immediately try to relieve from the allergic symptom. And there is a severe form of epinephrine or steroid. You take it to neutralize this. But in the long term, you can manipulate immune system to help you become, resistant to allergic diseases. Anyway, just I'm trying to tell you that how this works. So the IgM be produced if the cells are making this type of cytokine. And so this is called isotype switching, which is very important from the disease standpoint. Okay, I know it's 10 past. What else I want to tell you? I feel bad that we don't have enough time to talk about all of those interesting part. That's the antigen presentation and processing. This is the cooking method, right? I mentioned. Then complement. Complement is another pathway which is extremely important, right? Complement mediated pathways, and complement plays important role in the innate immunity, but also in the adaptive immunity. When is that antigen antibody complex plays a role. And I think from the previous speaker you saw that terminology C3b. You heard about that C3b? Anybody caught that slide? Yeah, complement C3b, which is called opsonine. Anybody heard about the terminology called opsonine? Okay, opsonine is like a tag, right? So in the school you guys seen that sometimes people or kids put a sign, kick me, or you guys done that when you were in the elementary school? You guys had that you guys did that, right? They put some sign in the back and everybody is coming and kicking you. I'm not joking, right? It does that, right? But you have no knowledge that somebody put a sign in your back saying that kick me, right? And so your friends come in and kicking in your back, then you have no idea why they are kicking me, right? You see this? So that's when the kids become real nasty. So that's mean, right? Yeah, you play that game. So why they did that? Because there's a sign there, right? So C3b to me is like that sign. So what does happen? The C3b produced during complement activation, and this go and bind to the bacteria. So this makes the bacteria a target. Like if somebody put a sign on your back, you are a target right now, right? Are you getting it? You carry some kind of signal that you are becoming a target, somebody is going to come and kill you because you are the target. And so the immune system does that. And he used this molecule, C3b is one of those molecules produced, and this molecule come and bind to the pathogen or bacteria, and once it is tagged, macrophage know because it has a receptor for C3b, and then it go to bind and kill it. Same way antibody, same way a lot of those other molecules. So the complement play an important role that way. Okay. So anything you want to remember about complement? C3b. That's the one you want to remember. Complement is a cascade of enzyme. You have a one product, it get activate, and you can see this all this pathway. I can tell you all the names without looking at the slide, but for you guys, it takes a while. The C1QRS, we start with that, then become activated, then eventually we want C3b, and these are a lot of other molecules they are made in the process. And this is antigen, antibody makes it, but then the other one is called alternate pathway, where just the bacteria or virus, they can also activate this pathway. And this works very well though. This is really important pathway that's been used. And again, this pathway is for innate immune system, but it's not the previous one. The classical pathway is for antigen antibody complex. Okay. Okay. So eventually, I didn't get. So we talked about. I think the previous speaker mentioned about little bit highlighted how do we immune system help recover or control the biofilm, right? I mentioned that. I think that's the baseline is because it's a multi-layered, immune system cannot grab them, right? You cannot control it. And so you need to disperse them. You need to make it planktonic, and then immune system can work, right? And so other thing I mentioned that they become chronic diseases, right? And so eventually you'll find that a lot of those diseases become chronic because how the immune system works, right? And here also the planktonic versus biofilm, and same macrophage trying to control it, but then it cannot, right? Because of the multi-layer formation. I will stop here. I know everybody has a limit, right? And I'm very sensitive about limit because I can talk forever, but doesn't mean anything to you if it is not passing your screen in the front, right? So ask me any questions you have, because I'm trying to finish the immune system today, then we'll talk about the other. Tomorrow I'm going to talk about actually probiotic, one of the bioengineering aspects, which you should listen. It will be very funny part, because these are our actual experiment we did, and I'll tell you some of the stories behind making that bioengineered probiotic and what is the future of it. But before that, if you have any questions, go ahead. Oh, haptens and adjuvants, I didn't talk about that. You want to know what is hapten? Yeah. So hapten, you heard about hapten? Hapten is the molecule which are not generally antigenic. It's a small, right? Peptides, a lot of those, and the immune system cannot recognize them. Hapten. This could be not the presentation. Presentation you start with big, they will choose one. That's what they present it. But hapten, anybody working with the peptide, small peptide, antimicrobial peptide, bacteriocin, heard about it, right? And those are usually not immunogenic. I gave example of lot of antimicrobial peptides, small peptide, and peptide alone cannot induce immune system. So how do you make them antigenic? That's the question, right? So hapten, usually what you do, you tag it with the bigger protein, okay? And once they are tagged with the bigger protein, I think I have some pictures somewhere, and once we tag it with the bigger molecule, then you inject it, then you will produce antibody. And there is a chance that antibody or immune cells can recognize the hapten alone, okay? So that's how we develop antibody against small molecule. What is the practical application of hapten? Thank you for bringing that up. Why we need to know about hapten? I know besides doing research, right? You have a small molecule, you want to generate antibody, and you say generally we take bovine serum albumin, BSA, which is about 66 kilodalton. You combine it, conjugate it, or KLH, keyhole limpet hemocyanin. These are type of molecules you conjugate and you inject into animal, and that's what they make the antibody. What is the practical application of it besides the research? When you go to see a doctor for infection, any kind of diseases, that doctor asks you anything about, are you allergic to certain drugs? Do they ask you? I know many of you may never been sick, so maybe you didn't have that disease, but this is one of the common questions. If you go to hospital or US anywhere or see any doctor, first question, are you allergic to any drugs, right? Why? Why you have to worry about? Because allergic diseases are life threatening, right? So what's the relationship with the drugs and allergic diseases? Are you getting my question? They will ask you, are you allergic to any kind of medication or medicine? So the medicine can cause allergy. One example is, you heard about penicillin? Penicillin, they will ask you, are you resistant or are you allergic to penicillin? Why? What happens in real life, penicillin like hapten. Penicillin is a chemical structure, small molecule. It's not even protein. Okay? But how does it induce immune system? Why it become allergic? Why do you suffer from drugs? I'm using penicillin as an example. But penicillin like molecules also can induce immune system, make you allergic. Why? Come on, this is the part of the learning. I just said that. I told you the answer. So when you take penicillin like drugs, small molecules, what happens that in our serum we have a lot of proteins, right? And this penicillin, when it gets to the blood circulation, it can form complex with other proteins, and as a result it forms a larger molecule. Okay, and the larger molecule behave like an antigen, and now your body going to produce antibody against that complex, and that's so scary. You see this? Not everybody allergic to penicillin, because each time I go see doctor for any reason, just for health checkup, if they want to give me any medicine or even routine stuff, they will ask me, am I allergic to any kind of drugs? And that first reason they ask, because if they are going to prescribe anything anytime during emergency, maybe you are subconscious, right? They are going to give you some drugs, and they need to know, are you allergic to penicillin or are you allergic to any kind of diseases? So for your own sake, okay, here is the deal, that penicillin like molecules, they form complex with other proteins, and inside the body they are going to serve as a hapten, and now they are going to produce antibody or immunoglobulin, and now your body can respond to that. That's what makes it a very serious situation, because our doctor treating you, when in return you became sick because of the allergic response. So hapten is that one. Adjuvant, you asked for the adjuvant as a terminology. So the vaccine, whenever you introduce any kind of antigen, we add it called adjuvant. Adjuvant, purpose of the adjuvant is that it can activate immune cells. Okay, when they inject it, there is some other molecules like peptidoglycan. Peptidoglycan is a good adjuvant. So what it does, when they are introduced, they can recruit immune cells, and so when the immune cells come in, they will take the antigen or the vaccine, then they will process it, they will present it to the other immune cells. You see this? So the adjuvant is the way of activating your immune system by external means. There are certain molecules, and that areas of research also very important. Adjuvant plays an important role. Any other questions? Role of the CD molecules. Yeah, so CD is called, I didn't go through this, this CD4, CD8, all of this terminology, whenever you see it, CD called cluster differentiation, CD. And so the T cells, only OA or B cells. I will not present so much about B cells. B cells are easy to recognize because they make antibody. Okay, the T cells or macrophage or dendritic cells, all of those neutrophil, all of those cells has a certain chemical marker on their surface, and those are designated as CD, cluster differentiation. So if you see the 4 CD4 and or CD8, you saw this, the CD4 means the molecule is called CD4 because it reacts to that, and it's a marker for T helper cell, and CD8 is a marker for cytotoxic T cell, the T cell which kills the pathogen infected cell, the virus infected cell, cancer cells, they are actually attacked by CD8 cells. And so CD, anytime you see this CD designation, whether it's 11 or 78, 65, 45, all of those different designations, that actually help you to identify what type of immune cells are there. That's what the CD. Number stand for, it just help you to define that whenever you find CD4, we call them, these are T helper cells. So I think just, let me just, there's a table there. I think I close it, that'll be easier maybe. Oh, here in this table, so like CD1, the T cells you find in thymus, and CD2, all T cell has this. CD3 you will find it, all mature T cell has this. CD4 usually is a T helper marker, okay, and CD8 is cytotoxic T cells, and whenever you see CD4, CD25, FOXP3 is the transcription factor. These are called regulatory T cells. Okay, so these, all those numbers means they are certain purpose. Like if I say CD11 plus or plus, usually this called dendritic cell has this marker. Okay, this help us to identify what are those cells. So over the many years of research, people found out that these CD molecules can help identify what type of cells are playing a role, certain diseases or gut immunity or any aspect of the disease or any kind of response, immune response. So this is how they identify it. Okay. Any other questions? I know it's late, right? Okay. So I know I'm not going to talk much about immunology again, because again other stuff we need to finish it. Tomorrow when I talk about the probiotic, I will be presenting you a little bit of immune system, but because that's the immune modulation, we want to modulate our immune system. If you want to stay healthy, think about it. I said immune system is everything. What you are, your immune system, how well it keep you protected against whether disease or any kind of threat, right? That's why I always want to know how can I modulate my immune system? How can I help improve my immune system? Number one, exercise. Do you believe that? Exercise can help you immune system active. Then of course diet, that also help you. So number of factors help you to maintain your immune health. Okay, there are a lot of different ways people try to maintain their immune health, but then a lot of times you don't have any control. So autoimmune diseases, right? Some of you discussed about autoimmune disease. Your immune system attacking your own cells. There you can't control it, right? Only way that you have to suppress your immune system, right? During surgery, certain organ transplant, we have to suppress our immune system to accept the organ. If you are going through heart transplant or kidney transplant or liver transplant, so sometimes you are getting from somebody and your body may reject it. How do you do it? You suppress your immune system so it is maintained for a while. Okay, so immune health is extremely important. To me, I think it's everything. Okay, and how do you keep it active? That's very important. Okay, I know, so I will not be able to talk much about it. I know I had a chance to to give you just the gist of how it looked like. But then go over the slides, ask me any questions during these next 2-3 days. I'll be able to help you. Any quick questions? No? Okay. Class dismissed. So feel free to leave and relax and enjoy this evening. Okay. I will see you tomorrow morning. Probably we'll start with the antibiotic resistance. We'll do the survey a little bit, and then we'll talk about the probiotic, or especially the